Hola, muy buen día para todos y buenas tardes para aquellos que nos acompañan desde Sudamérica y Europa. En nombre de la Asociación Mexicana de Especialistas en Nutrición Animal, AMENA, les doy la más cordial bienvenida a esta nueva faceta de actividades en línea, que afortunadamente ha sido una excelente válvula de salida para desarrollar los objetivos de la asociación, como son, entre otros, la capacitación, la interacción, el intercambio de ideas y conocimiento con los especialistas involucrados en la nutrición animal y en actividades afines. Dado que este año tampoco será posible llevar a cabo nuestro congreso acostumbrado, incluyendo el 9º Congreso del Colegio Latinoamericano de Nutrición Animal, CLANA, hemos escuchado la opinión de nuestros socios y tomado la decisión de seguir avanzando en formatos más completos de comunicación. Y es así que se concluyó en organizar esta serie de simposios mensuales en donde discutiremos con mayor profundidad temas específicos. Entendemos que de una u otra manera, a pesar de las restricciones de movilidad, problemas de salud y cualquier otro que tengamos presente en estos momentos, el trabajo diario lo tenemos que seguir desarrollando. Y por eso dábamos inicio a los seminarios a las 5 de la tarde. Pero como no queremos perder la oportunidad de tener interacción con especialistas de todo el mundo, hemos adoptado este nuevo horario de las 10 de la mañana a la 1 de la tarde. Eso sí, solamente durante un lunes de cada mes, lo que nos permitirá tener tiempo suficiente para otras actividades. Afortunadamente también la industria nos escucha como siempre y nos ha apoyado con sus patrocinios, por lo que estamos muy agradecidos con las empresas participantes. En esta ocasión, Avevista, Adiseo, ADM, DSM, Elanco, VFarma, Nuproxa, Novus y Provimi. Quiero hacer un reconocimiento especial también a nuestros patrocinadores platino, ya que nos van a estar apoyando durante todos los simposios. DSM, Elanco, Nuproxa y Provimi. Asimismo, extiendo la invitación a todas aquellas empresas que deseen participar en el patrocinio de los simposios siguientes. No olvidemos que la MENA es de todos y funciona gracias a los apoyos que recibe de los socios y de la industria, lo que permite realizar estos simposios sin costo alguno para los asistentes. Así que aprovechemos esta oportunidad para difundir entre los colegas, amigos y clientes estas actividades que hoy damos inicio. Agradezco a todos su asistencia y aprovecho también para agradecer a los coordinadores responsables de este simposio, el ingeniero Rogelio Elizarrarás, el ingeniero Silvestre Chárraga y al doctor Sergio Fernández, quien además será el moderador de este simposio. Sergio, ¿puedes por favor encender tu cámara? Sergio tiene un doctorado en nutrición de aves en la Universidad de Illinois. Actualmente trabaja como asesor de nutrición avícola para Elanco Animal Health. Sergio, la conducción de este primer simposio está en tus manos. Gracias nuevamente por aceptar esta invitación.
Muchas gracias, señor presidente. Bueno, muchachos, hoy tenemos un simposio excelente. Los tres ponentes que tenemos son de primerísima línea. Vamos a tener las tres pláticas en el orden en que aparecen aquí, el doctor Bedford, el doctor Cowieson y la doctora Ángel. Y vamos a tener 40 minutos para cada quien. Y las preguntas, de favor, les ponen por escrito en la sección de preguntas y respuestas que aparece hasta abajo en el Zoom. Y durante la sesión al último vamos a tener una mesa redonda. Ahí se van a leer las preguntas y los ponentes van a contestar. Entonces, voy a proceder a comentar sobre el primer ponente. El primer ponente es el doctor Mike Bedford, que tiene un bachelor de la Universidad de Nottingham en nutrición animal. Después tiene su maestría en la Universidad de Guelph en nutrición también, y un doctorado en la Universidad de Guelph en nutrición y bioquímica. Sí, el doctor Mike Bedford tiene muchísimas publicaciones y precisamente de lo que nos va a hablar hoy es de lo último que estaba estudiando, porque él es un experto muy grande en el área de enzimas, en el área de avicultura, y ha estado trabajando mucho en los efectos de las enzimas como prebióticos. Entonces, sin más, los dejo con el doctor Mike Bedford. Mike, please, could you please turn on your camera and your microphone?
Many thanks, Sergio, and thank you very much for inviting me here. I'll just share my screen so that you can see my presentation. Thank you, Sergio, for the introduction, very kind introduction, and I'm delighted to be here. It's just a pity we're not here in person, but nevertheless, these kinds of interactions are always very stimulating and useful for all involved, and I hope this presentation will bring something of interest to you all. My topic today is the influence of enzymes on intestinal health, and specifically I want to focus on the area of nutrition of the microbiota, and how that influences whether the animal has a good outcome or a poor outcome in terms of intestinal health and structure. So if we think about it, intestinal health is governed by a number of facets, principally the microbiota, the species and the activity of the microbiota in the gut, which has an influence on inflammatory responses and the way the immune system responds to these challenges may actually have a greater impact on the animal performance than the initial insult of the disease itself. But with respect to what we can do from a nutritional viewpoint and from an enzymatic viewpoint, there's been quite a bit of work recently, and in fact since the early 90s, looking at how enzymes and how nutrition can influence the structure and activity of the population of bacteria that live in the gut. And the structure and population is governed by nutrient supply to both the ileum, and I'll speak about the ileum first, or the small intestine, and then later I'll talk about the cecum, where some of the bigger problems originate, but then often migrate back towards the ileum itself. And when we talk about nutrient supply, we're interested in multiple nutrients here. We're talking about starch, protein, fats, and fermentable fiber in particular. And fermentable fiber obviously is something of great interest to us more recently with some of the recent work that's been going on with fiber structure and interactions with oligosaccharides. So moving along, if we think about the ileum, this is an old slide, but it's still as valid as it was when I first put it together. It must be 20 years ago. If you have a highly digestible diet and the animal is healthy and the gut is functioning well, then you'll see that the starch, the fat, and the protein is very rapidly digested. Hopefully you can see this pointer, yes, rapidly digested and absorbed. And there's very little nutrients found lower down in the gut, in the terminal ileum, in the last third of the ileum even, where the pH and the anaerobic structure of the environment is favorable to many pathogens taking hold. So we can limit the amount of nutrients arriving here, then we will limit the populations of bacteria that can grow. We can do this by feeding highly digestible diets. We can improve this situation by feeding enzymes, and that's the first point. The first point I'll be mentioning enzymes, but many times throughout this presentation I'll come back to that. Now, clearly, if things are going wrong and we have significant amounts of material being undigested, the fat and the fiber and the starch, protein getting down to the back half of the small intestine, then you create a substrate overload and you start to see really significant increases in the microbiota numbers in this section of the gut. These start to compete with the host, and as a result you'll start to lose performance. And there's some really nice data I'll show you from the 1990s where this even turns into significant amounts of starch being fermented in the ileum. So if we think about it from the viewpoint of an animal as it's going through the stages of growth, when very young, at point of hatch, the digestive system is set up such that it's not particularly well adapted to digesting all the nutrients which it is being given. The piglet, for example, is pulling from milk onto solid foods, and the chick is going from material absorbed through the yolk and the yolk sac and then going straight onto solid feed, which is far less digestible and easy to absorb. And the diet that we give them consists of a number of nutrients, which are listed here on the left-hand side. So we have monosaccharides, we have starch going all the way down to cellulose present in your diet in the form of structures and nutrients present in all the ingredients that you feed. Now, monosaccharides are very well absorbed and well digested, so they'll have gone by the time this material in a young animal has cleared the small intestine. So very little glucose will appear in the large intestine of a chicken, and very little will appear in the feces. But as you go down this list of nutrients, what you'll see is they become progressively harder to digest and harder to ferment as well. So we start fermentation of fructans and go all the way down to cellulose. You can see fructans are relatively well absorbed, well fermented, and as a result, their energy is released. And then we get into fructooligosaccharides, beta-glucans, mannans, pectins, xylans, and cellulose being very indigestible. So as far as a young animal is concerned, the nutrients which are of most use to it are here, and the nutrients which are of interest to the microbiota are all of the rest. And notice it includes starch and protein, and always will include starch and protein. And it's this that's got to be considered when trying to generate a very healthy gut and understanding how enzymes can help. As the animal ages and its digestive system becomes more effective and its microbiota become more effective at fermenting harder to ferment materials, then you start to see materials such as mannans and galactans, pectins, and xylans start to be fermented in the large intestine, and there's some energy value coming out of it. And you'll notice that starch and protein is less available to the large intestine, as digestion in the small intestine is complete, almost complete. And then in the adult, we see a situation like this. And really what I'm trying to build here is, over the period of about 28 days post-hatch, the chick is exposing its large intestine to an ever-decreasing range and quantity of nutrients for its cecal or large intestinal microbiota. And I've tried to summarize that in this slide here, which is really taking digestibility data and saying, well, what was not digested, and so therefore what is available in a 2-day-old, a 5-day-old, and a 10-day-old chick. And what you can see is, well, starch and protein to begin with are not particularly well digested. So there's a large percentage of the starch and protein which does pass through the large intestine and is potentially a substrate for the bacteria. As the bird ages, starch and protein drop, but they're still present. And what you can see is that the importance of other fiber fractions as being the carbohydrate that can be fermented in the large intestine take a greater role of import. So they become more important as sources of energy. It's not ever going to be the case that cellulose is going to be quantitatively fermented, but xylan, which is the next most quantitative fiber source here, as shown in the green bar, is a substantial amount of potentially fermentable material, which is really important to understand when you consider this transition. So as far as the bacteria in the large intestine are concerned, a lot of easy to ferment material here, it's depleting here, and it's almost gone here. So over a very short period of time, the microbiome in the large intestine have to adapt to the removal of some very easy to ferment nutrients, such as starch and protein, and adapt to ferment what's left, which is the fiber sources, which will get down into the large intestine, which ostensibly is the xylan, the mannans, galactans, and pectins. So how does this play out in terms of what happens to the bacteria that actually succeed to inhabit the gut of a chicken over its lifespan? Well, it's shown quite nicely in this piece of work, which just simply shows from 2016 work by Baloo. If you look at what are the dominant species here in pink, proteobacteria, gamma proteobacteria, this is day 0, day 1, day 3, day 7, day 14, and day 28 of age. You can see there's a significant shift where these bacteria in pink are being replaced by Firmicutes clostridia, which are fermenters. So you're replacing bacteria which are using protein and starch sources ostensibly, and replacing them with bacteria which are now looking at the fiber as their main carbohydrate source and source of energy. Now, this has been shown quite nicely to be easily disturbed if we have poorly digested diets. And some of the work that was done back in the 90s using very sticky, viscous gums as a means to slow down digestibility of nutrients. In this case, it was particularly starch. What you can see here is this is control birds Starch digestibility, the 21 to 29 days of age is about 90%, and protein digestibility about 80%. And ileal VFA concentrations in these control birds fed a sorghum soy diet was quite low, about 8 to 10 micromoles. They fed an NSP fraction, which is a viscous NSP fraction and very sticky, slowed down digestibility of all nutrients, and as a result, starch digestibility dropped to below 60%, and protein digestion fell to almost 50%. And as a result of this material not being digested by the animal, the ileal VFA concentrations increased dramatically to 120 micromoles. So you can see the lack of the ability of the chicken to digest the starch and the protein meant there was far more substrate available for the bacteria to ferment, and as a result, the volatile fatty acid levels in that part of the intestine increased. And that's clearly not beneficial for the animal viewpoint, not just because of loss of nutrients, but because some of the concentrations found of lactic acid, for example, can be quite damaging to the gut itself. And in this particular treatment here, what they did, they fed the same viscous NSP, but they fed an enzyme which simply depolymerized it, and in doing so, enhanced digestibility of starch and protein again, and reduced the VFAs in the ileum dramatically. So if we clear the digestive constraints out from the diet, if we enable the animal to have a highly digestible diet with no constraints on digestion, then the amount of fermentation in the small intestine can be minimized. And that is something we should strive towards, as what we want is starch and protein to be used by the animal, the host, and not the microbiome itself. Now we turn to the seeker. And what we have here is a different situation as well. We have undigested starch, protein, fat, and we have fiber that can go into the seeker. Now, protein is labeled red here because this is the alarm button, if you like. This is something you do not want to get into the seeker, and we'll go into this in a little bit more detail in a moment. So starch, protein, and fat can enter the seeker if they've not been digested well, as I've just mentioned, and then fiber. In order to get into the seeker, it has to be soluble. And if it is insoluble, it has to be very, very small particulates. It will not get through the cecal sieve if it's of any size whatsoever. So if it's soluble, there are some things to consider as well. It's not just whether it's soluble or not will make it fermentable. It's how big it is, the molecular weight. Is it an oligomeric? Is it a mid-sized polymeric? Or is it high molecular weight and viscous? And as you get from oligo to mid-size to high molecular weight, it becomes less and less easy to ferment that material. The sugar composition of the backbone makes a difference as well. So whether it's a xylan, a mannan, a galactan, it doesn't matter just saying you have a large molecular weight fermentable fiber source. If you don't know what the makeup of it is, you don't know how easy it'll be fermented by the bacteria that inhabit the seeker. And then substitution. Now, what do I mean by that? If you have an arabinoxylan from corn, it's generally more highly substituted than that from wheat. And what that means is there's more arabinoses on the xylan backbone, and that just slows down the ability of the microbiome to break it down and to produce fermentable material from it. And as I said, the insoluble material has to be very, very small to get in, but it could be play a critical role in providing source substrate and energy for the bacteria as time progresses. So as I say, the challenge is to limit protein flow to the seeker, and it's pretty nicely explained in the Appiolati paper of 2014. Basically, bacteria which have the ability to break down both carbohydrate and protein, so saccharolytic or putrefactive metabolic potential, will always preferentially use the carbohydrates. So it would rather be saccharolytic than putrefactive, and it will always therefore use the carbohydrates if they're there. So that's the first important point to make. You've got to recognize that one of the problems is not just how much protein is available, but also how much carbohydrate there is there as the alternative to attract the bacteria away from the protein so that they can produce something beneficial in the form of volatile fatty acids rather than putrefactive metabolic products. And putrefaction is usually intensified towards the distal intestine, and that's even true probably within the ceca. If you think of the ceca being in the distal sections of the intestine, where the carbohydrates may become depleted towards the end of the day, and protein is the only material that's left. So we need to consider items of position in the gut as well, and timing of release of carbohydrates. And so we need, ideally, we need fibers which keep carbohydrate fermentation going throughout the colon and the ceca, and suppress, as a result, fermentation processes. Just want to show you something that can really mess up digestion, is if, for whatever reason, protein digestion is compromised. And in this particular study, what we did was we fed an antacid. It's actually a proton pump inhibitor, which dramatically reduced the ability of the chicken to produce acid. And you can see this quite clearly here. There's the control birds with an average pH in the gizzard of about 1.8. And in the birds fed the antacid, the pH was all the way up at 4. Now what this does, it prevents pepsin from really doing a very good job at breaking down protein as it comes through the gizzard, proventriculus gizzard. You can see it didn't have a massive effect on pH in the ileum or the cecum. So the major effect here is in the gizzard. You can see the pH, as I say, almost doubling from 2 to 4. Now, what were the consequences? Well, in the control-fed birds, ileal nitrogen digestibility was 60%, and that fell to 43%. So quite a significant reduction in nitrogen digestibility. And that resulted in a lot more soluble protein being seen in the ceca. And then the products of protein putrefaction, here seen as indole and P-cresol, are significantly enhanced when we fed the bird the antacid. So the fact that protein digestion has been compromised has resulted in a huge excess of protein going into the ceca, means that the bacteria that can use protein and ferment it and actually putrefy it have got a lot of material to work on, and as a result produce a lot of these products. And these products are actually quite detrimental to the structure of the gut and the health of the animal. So how do we limit putrefaction? Well, we can limit protein flow. So I've shown you how not to do it, which is by feeding it antacid. But in this particular case, limiting the protein flow, we can use one of several enzymes. In fact, many enzymes that are used in the market today have an influence on amino acid digestibility. And I'm not going to go into the details of them all, but NSPases, proteases, and phytases all have a role to play in limiting protein flow into the large intestine. And then what we can also consider is maximizing soluble fiber provision as an alternative, or as an additional activity as to limiting protein flow. And we can do this again by feeding NSPases, and I'll show you some data that we've generated recently to give you this as a concept. We can feed oligosaccharides, which are not digested by the animal, but are very much so fermented by the bacteria that inhabit the large intestine. And we can actually use a combination of these two. So we could be looking at using a xylanase, an NSPase, a glucanase, and an oligosaccharide in combination to optimize delivery of fermentable carbohydrates. So I want to go straight to the concept that we think is working, so you can understand the next few slides to describe what we think is happening when we're feeding NSPases and oligosaccharides and using this combination, what's called a symbiotic mechanism. So a little bit of a complicated slide, but this is the original this is an adaptation of the original prebiotic mechanism that was first discussed way back in 1996. And the original idea was that, well, we feed enzymes which break down cell walls and they produce oligosaccharides here. And these oligosaccharides are then fermented and produce VFAs, which give energy to the animal. But what we're realizing now is that the amount of oligosaccharides that we produce when we feed an NSPase is very, very small indeed. But it's enough to change the activity of the fiber-degrading bacteria who survive, who live in the ceca, such that they produce their own enzymes. And I've said xylanases here, but they're producing more cellulases, they're producing more mannases, they're producing more fiber-degrading enzymes in general. So these oligosaccharides are acting like a signal. They signal the bacteria that inhabit the small intestine, the large intestine, sorry. They produce lots of their own xylanase, which break down the cell walls, break down the soluble material into smaller oligosaccharides, which are then fermented into these different volatile fatty acids. They allow the microbiota to focus on fermentation of fiber rather than switching to protein. And as a result, the host gets more energy. And actually, there is a feedback mechanism from the ceca where the butyrate in particular is picked up by L cells in the ceca, feeds back to the gizzard, and actually stimulates longer holding time in the gizzard, giving you better protein digestion. So that's the theory. That's what we believe is happening now with the use of xylanases combined with oligosaccharides. And now what I want to do is just go through some data which hopefully will explain how we've come to that conclusion or that idea. Firstly, let's have a look at what happens when you have the availability of a fermentable carbohydrate in a situation where there is production of putrefacted products. So this is a simulated, this is swine colon simulated in vitro. And what we've done is we've incubated controlled swine colon pigs with 1% polydextran or xylans, so fermentable carbohydrate sources. And what you can see is there's an increase in gas production and an increase in total VFAs, suggesting that there's greater activity in this colon of these these microbiome that are growing ex vivo when we give them more substrate to ferment, which is evident. It's not surprising at all. So we'd expect them to ferment and produce more volatile fatty acids. But what was interesting was by supplying carbohydrate, what we did was actually by feeding the 1% PDX or the xylan to these in vitro ex vivo simulations, we're reducing indole and scatol production, which are breakdown products of tryptophan and phenylalanine, and we've got pH reductions as a result of VFA being produced, and branch chain VFAs, which are directly produced from leucine, isoleucine, and valine fermentation. Their concentrations are reduced. So it shows that if you have more fiber to ferment, more soluble fermentable fiber to ferment, you will limit the amount of putrefaction ongoing in the large intestine. Now, do we produce oligosaccharides? Do we produce material when we incubate different substrates with our enzymes? Well, this is some work by Bang So et al. Looking at a product called RoboBioXL or Advance, actually XL2 is Advance. And they were looking at whether they fed or incubated the whole grain of wheat. So this left-hand panel here is all wheat. Being incubated with just a blank, so a control, or enzyme 1 and enzyme 2, and allurone of wheat, so the allurone fraction only or the bran fraction only. And what it's basically showing is that when you incubate wheat with their enzymes, you increase the amount of xylose release, which is soluble xylan. So they're increasing the release of insoluble into solution. It's not telling you what size it is, it's just telling you you've made more of the insoluble soluble. And as I'll show you in a moment, a lot of it will be high molecular weight material. If we go over to maize here, you can see that the incubation with both the NSPases has increased the amount of soluble xylan being released. So not quite to the same extent that you see with wheat over here, but even incubation with maize, we are getting soluble NSP release from maize when we incubate with the enzymes. Now, that's total. What about oligosaccharides, the bits that I was talking about, the DP2, so xylobio, xylotriose, etc. That's what we're interested in, really, from the stimulating the bacteria, signaling them to actually give us more xylanases themselves. So again, on the left-hand panel here, we have wheat. And this is an accumulation of the blank, and this is enzyme 1 and enzyme 2, incubating with whole grain wheat. And you can see there was a significant increase in the release of oligosaccharide arabinoxalan. So this is material which is up to about 4 to 6 xyloses long, very short, and these are the very same oligosaccharides that I mentioned earlier are being used as a signal by the bacteria inhabiting the ceca for them to produce their own xylonases in a much larger quantity. So it's very evident that you can see significant quantities of these oligosaccharides being produced from the different fractions of wheat when incubated with these enzymes. But when you switch to maize, you can see very clearly, it doesn't matter which fraction, there's very little oligosaccharide being released from maize. And that's one of the biggest differences that we've seen between maize and wheat. Yes, maize is much less susceptible to xylonases, that's true. But the biggest difference is it releases virtually no oligosaccharides. So you don't get this signaling effect very clearly set up when you're feeding maize compared to wheat. And that's why we believe that you get more response when you feed a xylonase coupled with an oligosaccharide, simply because the xylonase, when fed to the maize-based diet, maize-fed broilers, will produce free and soluble arabinoxalan, but it will not produce the signaling oligosaccharides. And it's important to actually see where these oligosaccharides have their effect. This is a paper that's actually in production, but it's actually from Tom Dale's thesis 2019 out of the UK here. And what this was showing was different concentrations of xylose, xylobiose, xylotriose, and xylotetraose in different parts of the gut. So the jejunum, the ileum, the colon, and the cecum. Now, these are very small graphs. And what I want you to just simply view and see is that when we look at xylobiose, which is X2, so there's 2 xyloses there. This is a very small oligosaccharide. And in vitro, this is highly used by lactobacilli and bifidobacter. You can see that the concentrations in the ileum and the colon are lower than in the jejunum. And it doesn't matter whether we're feeding our product, this is xylonase here, it didn't change the concentrations of xylobiose in these sections of the gut. But the most interesting point is this concentration of X2 reduced as we went from the jejunum to the ileum, suggesting that it is being consumed dramatically, because you'd expect it, if it's not being absorbed by the animal, it should increase in concentration, and it's not, it's actually going down. So this is actually being utilized by ileal bacteria. And you can see with xylotriose, it's not going down in concentration, but it's not going up either. So there is some consumption of X3, and with X4, if anything, there's a slight increment. So it seems like the shorter the oligosaccharide, the more proximal that oligosaccharide is going to be fermented and utilized by the bacteria that inhabit the ileum. They all get used in the ceca. You can see they're all disappeared the minute you move into the ceca, very highly utilized and very quickly fermented. And the important point here really is that when we tend to think of oligosaccharides and fermentation being confined to the ceca, but clearly there's something we've got to consider in the ileal phase as well, and it's often overlooked. And these are some of the latest pieces of work that we're looking at now, whether we need to target different types of oligosaccharide for the ileum compared to that for the cecum. This is some work that it's just been published, published in 2017 now, and it simply said what we're looking at here is the concentrations of soluble sugars that are found in the ceca. So what we've simply done is taken the contents of the ceca out, and we've measured the total amount of arabinose in red and xylose in green, and then other sugars here in different colors. And when we fed a control diet or one with an enzyme, you can see the enzyme, which is a xylonase, significantly increased the xylose and the arabinose content. So in other words, it increased the amount of arabinoxalan that went into the ceca. This is birds that are 10 days of age. 11 days of age. This is birds that are 21 days of age. And you can see again, the enzyme is pushing more arabinose and xylose into the ceca. It's breaking up the fiber. It's making it soluble. And that soluble fiber can now go into the ceca. And principally, the big difference between control and enzyme-treated birds is the arabinoxalan content in the ceca. Now, at 42 days of age over here, you can see that there's a very small effect. It's still there, but it's a very small effect. And this is actually, it's not suggesting that the enzyme no longer is pushing soluble arabinose and xylose into the ceca, but what's actually happened is between 21 and 42 days of age, the microbiome has started to use arabinoxalan very effectively, and there's much more of it in the enzyme-treated birds. So in effect, what happens is we get more VFA production. And that was shown quite nicely. We measured in the same birds VFA production. So this is the same 11-day birds, 21 and 42-day-old birds. And what you can see is at 11 days of age, there's no difference between those two treatments at 11 days of age. Even though we know there was a lot more sugar going into the seeker, there's not much difference between them because the capacity of that microbiome to ferment arabinoxalan is not present. Even at 21 days, you can see no effect, but at 42 days of age, we see a significant increase in the amount of acetic and particularly butyric acid in green. So what we have done is with feeding azalanase, we pushed more fermentable arabinoxalan into the seeker, and it's only in the older bird in this particular study that we've adapted those bacteria, the microbiome, to use the arabinoxalan. Now, the critical bit here is that in order for this to be effective, we need to move this quickly. We need to have the arabinoxalan degrading bacteria adapt and produce those alanases and ferment fiber as early as possible in the chick's life, so that it does not run into a situation where the seeker is suddenly starved of starch as its carbohydrate source and hasn't adapted to start using the xalan. This is some work that Juha and myself published in 2018, and it simply shows adaptation, what has happened over a period of time. So we had birds here that were fed a diet without or with xalanase. So the red is the control and the green is with xalanase. And we simply took the cecal flora out of those birds at 35 days of age. And we looked at how much gas was being produced from the control fed birds. And then we offered in vitro, this is all in vitro gas production, we offered this incubation, some NSP derived from wheat, so principally insoluble arabinoxalan, NSP from wheat treated with xalanase, so higher molecular weight, soluble arabinoxalan, if you like, and then arabinoxalan oligosaccharides, and then just xylose. So high insoluble, soluble, short soluble, and monomeric xylan material being offered to bacteria that have taken out of the control or a xalanase-fed bird's seeker at 35 days of age. And what you can see is a stark difference in the ability to produce gas, whereby those birds that have been fed a xalanase in the previous 35 days of their life have a microbiome which are much more capable of breaking down arabinoxalan than those birds that have not been fed a xalanase. So effectively, we have trained the microbiome to use arabinoxalan and use it effectively. So that's gas, that's telling you activity, and then this is butyrate concentrations, and you can see again a significant increase in production of butyrate in every single one of these cases, apart from in this case the axos. So we train bacteria to actually take active, if you like, become active and attack the substrates that are available in the seeker. And finally, just looking at some of the activities that we see in the seeker of the chicken. When we consider xalanase activity in the seeker, nobody's actually looked at, well, can you measure what's actually going on? Has anybody actually measured how much xalanase is there? And it has been done in the past, and we've been doing a lot recently. And we looked at the amount of activity of xalanase using the new Zal X6 assay, which is really useful for doing cecal analysis. It's a new method coming out of Megazyme. And what we can see is a very significant increase in the amount of xalanase that you can detect in the seeker when you feed a xalanase itself or a xalanase that is mixed with an oligosaccharide. And the really interesting bit is we added exactly the same amount of xalanase in these two treatments, but the addition of the oligosaccharide nearly significantly increased the activity of xalanase in birds at day 14, not so much at day 28. But you can see what looks like an apparent acceleration of the ability of that microbiome to produce xalanases by using a combination of a xalanase and a oligosaccharide. we don't just increase xylanase activity, but we also increase the size of the seeker. This is some work from Anne Bartil's thesis, where she was looking at just feeding Axos, so no enzyme whatsoever, but just the oligosaccharide, which would be signaling the microbiome to produce more xylanase and therefore degrade the xylem more effectively. And there's the control birds with a lighter seeker as a proportion of the total body weight. And the concept that Anne Bartil actually proposed from that work was that when we feed a xylanase, when we feed Axos, when we feed a symbiotic, as we were calling it, what we do is effectively increase the digestibility of total arabinexalam. So if we consider control birds, which are the hatched lines here, with birds just simply given the oligosaccharide, in this case there's no enzyme being given here, it's just the oligosaccharide, but you can see the total arabinexalam digestibility hits an asymptote here, maybe 2 or 3 weeks earlier than the control-fed birds, who take a much longer time to adapt and become capable of digesting the fiber fraction, the arabinexalam fraction of the diet. And this is the key, because if you can cast your mind back to the original slide where I was talking about what nutrients are available for the seeker to digest, to ferment, in this period here, in a young broiler, starch is being removed very rapidly, as are the fructans and the GOS. They're being consumed in the ileum. So it pays to have an auxiliary fiber source which is not going to be consumed and taken away from the bacteria in the seeker, so that they can produce volatile fatty acids rather than branch chain fatty acids or putrefacted products. So this acceleration of fiber degradation is really quite interesting, particularly in the area of antibiotic-free diets, where this is obviously becoming more and more key to very rapidly adapt a microbiota which is stable and is not producing deleterious putrefacted products. So I'm coming to the end of my talk. Basically the goal, what is the goal? It's to adapt the microbiome to the least digestible fermentable carbohydrate as fast as possible, and that's the xylans in this particular case. There are others, but quantitatively the xylans do sit in most diets: corn, soy, sorghum soy, and certainly wheat soy, as the most prevalent and fermentable and present carbohydrate that is present in the seeker. And how do we do this? Well, we need to supply the soluble xylan, obviously, so we've got to break material up and make it soluble, but we train the microbiome as fast as possible with oligosaccharides, and these oligosaccharides then enable the bacteria that live not only in the seeker but also in the ileum to attack the insoluble material and produce even more soluble fiber for their cohorts lower down in the gut. So the key messages I would like to bring home to this is a healthy intestine requires a balanced microbiota which preferentially ferments carbohydrate throughout its length and not protein. So we need to optimize the delivery of fermentable carbohydrate with passage through the large intestine. And remember, as things move through the large intestine, material becomes depleted, so it needs to be renewed. And the only way we can renew material for fermentation is to break down the insoluble material and release more soluble. And that's where the enzymes, I believe, are playing a very important role. We also need to stimulate the bacteria themselves to attack more difficult to ferment fiber, in other words, the arabinexalans and other such structures, earlier in the life of the chicken. And that involves NSPases, xylo-oligosaccharides, the arabinexalan oligosaccharides, and using them coordinatedly in a fashion to not just produce soluble arabinexalan, but the signal molecules as well. And then finally, don't underestimate the importance of delivery of undigested protein to the large intestine. And in that view, proteases, phytases, and NSPases all have a huge role to play in optimizing the delivery of carbohydrate to protein into the seeker and into the colon. So I'll finish there if I can. Stop sharing and take any questions if there are any questions.
Tenemos tiempo para unas preguntas. Voy a proceder a leerlas. Primera pregunta. Con respecto a los ácidos grasos volátiles, acético, propiónico y butírico son ácidos grasos volátiles. El ácido láctico no es un ácido graso volátil. ¿Es correcto esto, doctor Bedford?
Yeah. Lactic acid is interesting. We find far higher concentrations of lactic acid generally in the ileum than we will in the ceca, and it's generally arriving from fermentation of some easy to ferment substrates, where you can come from oligosaccharide as well. The interest in lactic acid is simply that if it does get into the ceca, which a lot will, and if it is being produced in the ceca, which it is, then there are lactate utilizing bacteria which principally produce butyrate from the lactate. But there's a very fine balance, and if you overproduce lactate, it has quite a low pKa, and it can change the structure and the function of the microbiome in the ceca and the ileum very rapidly. So it's a short chain fatty acid, but it's not a VFA, you're right, but it's an important one.
Gracias, doctor Bedford. Otra pregunta. ¿Cuál sería el contenido correcto de NSP solubles en pollo, pavo, en gallina reproductora?
Yeah, I think I need another 5 years before I can give you a definitive answer to that, and even then it might be not as definitive as you'd like. What I would suggest is I would be interested in limiting the amount of soluble in early diets and increasing it in later diets. We tend to do that where we're feeding wheat-based diets anyway. We feed much higher levels of wheat in older bird rations than we do in younger bird rations. But in some quantities, some types of wheat have too much soluble arabinoxalan, and that actually is a totally different problem. It's coming in from the viscosity content of it. But you can have over-fermentation in the small intestine. There's no two ways about it. Just to give you an example of what sort of levels of arabinoxalan would be nice to have, or soluble arabinoxalan would be nice to have. We did some interesting work in Hawaii, which has just been published, which shows that in a corn soy diet, you can dilute the diet 5% with wheat bran and even 10% with wheat bran, and you don't lose too much in terms of performance, simply because what you've done is produced a huge amount of fermentable fiber that is actually actively used when that diet was supplemented with the xalanase. So, and the interesting point was, you could see it very clearly in the VFA effects. The xalanase had very little effect on VFA production in the corn soy diet, but it had a huge effect on the VFA production in the diet containing even just 5% wheat bran. So consider 5% wheat bran as something you might want to consider, or something like that in the future, as being a substrate to kickstart a microbiome which is healthier and more capable of using arabinoxalan as a substrate and producing volatile fatty acids later on. So it's not going to be a huge amount of soluble NSP, but actually at the moment I don't think I'd like to put a number on it yet. Notwithstanding, the fact is that we don't know fully yet. When we talk about soluble NSP, we need to actually start to break that down into soluble arabinoxalan, soluble mannan, soluble galactan, but then we need to break that down into the size of that soluble NSP. So is it oligomeric, is it polymeric, or is it huge and viscous? And then the sugar composition comes into play as well. So they're some of the refinements that we need to work on before we can give definitive answers to that question. But it's a very, very, very good one to put out now.
Gracias, doctor. Siguiente pregunta. ¿Es cierto que las gilanasas rompen el efecto jaula de la fibra del alimento y de esa manera liberan energía?
Yeah, I mean, it's interesting. I don't actually have much confidence in the cage effect mechanism per se. And what I mean by that, I don't think the enzymes really, at the levels that we're adding, the concentration of the enzymes we're adding, the time that we're giving the enzyme to break the cell walls down, and the degree of destruction that is needed to release those contents, I don't think the enzymes have enough time to do that. However, it does happen in the animal. And I think the reason why it's happening in the animal has more to do with what's going on in the ceca than what's going on the enzyme breaking the cell walls down. And let me just elaborate on that. I mentioned that we enhance VFA production when we use these enzymes by producing more fermentable NSP in the ceca. That increases production rate of butyrate and acetate. Both of those VFAs have an influence on structure and function of the GI tract and feed back, particularly butyrate, through PYY, IGI, and lots of other enterohormones, feed back to the gizzard and delay emptying. And you only have to hold things in the gizzard for an extra 10 minutes and you'll get significant cell wall disruption, not only because of the grinding effect of the gizzard, but also because if you incubate cell walls in an acid solution, you will slowly break down the cell walls in any case. So my view is that the enzymes are working via, if you like, a cage effect mechanism, but not by direct depolymerization of the cell walls themselves, but through a mechanism which encourages the gizzard to do a better job of grinding and breaking down the material using acid and grinding activity to break open those cells and expose the starch and protein, which we do see by microscopy in the small intestine.
Muchas gracias, doctor Bedford. Vamos a parar aquí para tener un descanso. A las 10:55 comenzamos de nuevo. Y hay algunas preguntas que se quedaron en el tintero, pero las vamos a tener en la sección de preguntas y respuestas del final. Gracias. Bueno, bien, son las 10:55. Vamos a seguir con las pláticas. A continuación tenemos el honor de presentar al doctor Aaron Cowieson. Su patrocinio viene por parte de DSM. El doctor Cowieson tiene su maestría en química analítica de la Universidad de Aberdeen. También tiene su doctorado en nutrición avícola de la Universidad de Aberdeen. El doctor Cowieson ha publicado muchísimo en revistas con comité editorial acerca del uso de enzimas en avicultura. Actualmente es profesor adjunto de Animal Science en Purdue University y también es la cabeza de investigación de DSM Animal Nutrition and Health. Entonces, sin más preámbulos, los voy a dejar con el doctor Aaron Cowieson. Aaron, please, could you share your screen?
I'm just going to move you over here so that I can see my slides and make eye contact with you all at the same time. I hope that you can all see the slide. I would just like to say welcome to my home office. I think we're all struggling a little bit at the moment with all of the restrictions. I'm sure it's the same in Mexico. Thank you very much for the invitation to present this morning. So I've just got a few slides that I've prepared on the anti-nutritional effects of phytic acid. I've tried to keep it reasonably practical, and particularly toward the end, you'll see some of the applied part of the presentation. But I want to start the presentation just with one or two definitions, and a small amount of biochemistry. So please bear with me as I run through those points. So this is the overview of the presentation. We'll look at what phytic acid is, why it's important in nutrition. We'll look at the prevalence and variability of phytate in different feed ingredients, not extensively, but I'll mention a couple of points on that one. And then look at some of the interactive factors that go on with phytic acid. As you'll see as we go through the presentation, phytic acid is a very reactive compound, a very reactive molecule rather, and it's quite a number of adjacent factors that become relevant when we talk about phytic acid. And then I'm going to finish the presentation talking a little bit about the amino acid matrices that we use in phytase, where they come from and how they differ, looking at the comparative effects of phytase in swine versus poultry. Just one or two particular points on that topic. And that actually follows on quite nicely from what Mike was saying in the first talk about managing the flow of endogenous protein into the hindgut of animals. So just a couple of points on definitions, because we do get ourselves into some difficulties here. I hope this doesn't trespass onto Roselina's presentation, which will follow mine. I'm not going to talk about calcium and phosphorus extensively or really very much at all in this presentation, but just for the purposes of clarity, 1 mole of phytic acid is around 660 grams. And when you look at the contribution of phosphorus, it's around 1/3, around 30% of that, of 1 mole of phytic acid is phosphorus, and approximately 1/3 is mayo-inositol. And then the missing part is the oxygens and the hydrogens that are associated with the phosphorus. So phosphate, it's around 70%, 30% mayo-inositol, but phosphorus is about 30%. And then there's all of these different terms that you come across. Phytic acid, which is the fully protonated acid form of phytic acid. Then you've got phytin, which is typically an insoluble salt of phytic acid with different cations, typically calcium, potassium, and magnesium. Phytate, which is a term that's essentially synonymous with phytic acid. Phytate phosphorus is the phosphorus that's bound to phytate, quite logically. Non-phytate phosphorus is a calculated term where the total amount of phosphorus and the total amount of phytate phosphorus are simply subtracted one from the other. Digestible phosphorus, of course, we usually determine that directly at the ileal level, at least in poultry. via direct assessment of the disappearance of phosphorus relative to the ingestion of phosphorus in the feed. And then you have available phosphorus, which is the relative bioavailability of phosphorus, which would normally be compared against an inorganic phosphate source like dicalcium phosphate or monocalcium phosphate. Phytate phosphorus is usually present in diets around 0.24%. Under the microscope, this is what phytate or phytate globoids look like. So this is a nice paper, Bon et al. In 2007. And what they showed here is that the phytate crystal, if you see in the center of these spheres, you've got the phytin crystal, usually magnesium, potassium phytate, but they're surrounded by a protein shell or a globoid, which is partially resistant to phytase activity. The good news is that mostly that protein shell is solubilized when feed is ingested by the animal in the gastric phase of digestion, and then that phytate is solubilized into the lumen, into the digesta. So why are we interested in phytic acid as an antinutrient? So I've mentioned that it's present at approximately 0.23, 0.24% phytate phosphorus in diet. So it's essentially ubiquitous in all animal rations. But the antinutritional effects of phytic acid are associated with the charge on the molecule. And that's associated with the oxygen in those phosphate groups. And you can see here in this little cartoon, we've got the myo-inositol ring. We have 6 phosphate groups attached to the myo-inositol ring by ester bonds. And each of these different oxygens has different capacities to become negatively charged when the associated hydrogen is lost. Now, from a practical nutrition perspective, what that means is at pH of around 1 or 1.1, phytic acid, all of those oxygens will be protonated. They'll have a hydrogen ion associated with them, and therefore phytic acid will be neutrally charged. Phytic acid is never going to be positively charged, but it can be neutrally charged at very low pH. But as pH goes up, as the digesta moves from the gastric gut through into the small intestine and then into the large intestine, and pH is typically going up, hydrogen ion concentration is therefore going down, the propensity for these oxygens to lose their hydrogen ion and therefore become negatively charged increases. And you can see here that at the pH that we would find in the small intestine, phytic acid will often have around 6 negatively charged reactive oxygen groups that can then interact with positively charged nutrients like cations, positively charged amino acids and proteins and so on. So the whole anti-nutritional effect of phytic acid hinges on its electronegativity, and that in turn hinges on ambient pH. So low pH environments, phytic acid tends to be soluble, not particularly reactive, but at higher pH environments, it deprotonates, becomes increasingly negatively charged, and then it can interact with positively charged nutrients, compounds, and so on. I'm not going to go through this in detail, but you can see just a few examples of the phytate phosphorus concentration in some common feed ingredients. The point is, in cereals, it tends to be slightly lower concentrations. In protein meals and cereal byproducts, the phytate concentration tends to be higher. But it does vary, even within, as you can see here, within a cereal, within, let's say, maize, corn, there's quite a considerable range of phytate phosphorus concentrations. And nowadays, most of the phytase suppliers have NIR calibrations for measuring fairly rapidly the phytate content, and that allows more accuracy in feed formulation systems. The reason that phytate concentration varies is multifactorial, but It can be associated with genetics. You can see here, this is one example in the bottom left, different cultivars, in this case of peas, different growing locations, different phytate phosphorus concentrations. Phosphate fertilizers tend to increase phytic acid content. Here in this case, soybean, you can see on the x-axis of this frame, different cultivars of soybean with low, moderate, or high phosphate fertilizer applied, and you can see very nice response to total phosphorus and also phytate phosphorus in soybean. So we can see a number of different factors, the growing location, not only whether it's wheat or corn or soybean, but where that is grown, how much phosphate has been applied to it. So as nutritionists, then it becomes very relevant to understand the origin, the growing location of the soybean or the corn that you're using, and have some idea how much phytate phosphorus, what is the total phosphorus content of that material. I think this whole question becomes increasingly important when you are using higher levels of phytase, and particularly as people are moving to higher, more active, more bioactive phytases, the new generation of phytases, where phosphate matrices are often rather high, even approaching 0.2% available phosphorus. And then you really need to be sure, not so much whether the phytase is capable of generating that phosphate, but do you have enough substrate in the feed in order to allow that to take place. Even in corn, this is a review that Kurt Klasing and Hank Klassen and I put together a few years ago now, just looking at the IP6, so the fully phosphorylated phytic acid in just one ingredient, and even across, I think we sampled almost 200 different corns from across the Americas of Brazil, Mexico, US, and I think some Canadian corns. And even just within that one ingredient, quite appreciable differences from minimum to maximum in IP6 and also in IP5 concentration. So there is a variable substrate landscape out there, and it is important to measure these things, not necessarily all the time, but I think it's important, particularly if you change the origin of macro ingredients in the feed, to do a sense check, make sure that the phytate content is as expected. The other point is that not all phytate in feed ingredients is equivalent. And to be quite honest, I think this is an area that hasn't been explored as much as it possibly should have been. I think we're assuming that phytate from soybean, phytate from corn, phytate from distillery byproducts or wheat bran is essentially, the content is different, but the chemical properties of that phytate are similar. But actually the limited amount of data that we do have on this topic suggests that may not be the case. And you can see a couple of examples here, one from Rutherford in 2002, relatively old paper now, where you've got in the gold bars without phytase and in the green bars with phytase, how much phytate hydrolysis is taking place. And there was actually an ingredient by phytase dose interaction in this study, where there was fairly significant differences between the capacity for phytase to increase phytate hydrolysis under the conditions of this assay as you moved from wheat to corn, from rice bran to soybean. And then a more recent, well, actually not, it's 3 years before. This is another classic paper on this topic, Lesky and Kuhn, 1999. And here in laying hens, so this is now an in vivo study where the effect of phytase on phytate hydrolysis in soybean, in corn, and in rice bran was considered. And again, you can see here differences apparently in how accessible that phytate is, how susceptible it is to phytase hydrolysis. Estimates across the literature they vary, but somewhere between 50 and 90% of the total phytate is considered to be, in quotes, reactive or capable of participating in nutritionally relevant reactions in the intestinal tract. And other factors that will influence that include how much calcium is in the feed, what is the solubility of your limestone, the pH of the drinking water that the animals have, what about other cations like copper and zinc, their concentration in the feed. So it's a complex area, but I think we can make a really good start by simply measuring how much phytate we have, and then beginning to look at your calcium strategy, your limestone strategy, whether your drinking water may require to be acidified, and so on. So you can think about this in 2 stages. I put these slides together about probably 10 years ago now, so this is not a new slide, but I just want to, I think it's a helpful series of steps to help us understand how this anti-nutritional effect of phytic acid, which, as I described, comes from this negative charge that phytate has in the intestine. What is the cascade of consequences that occur once that phytic acid reaction takes place? So if you think about this, moving from the crop, you've got dietary phytic acid and dietary protein being ingested as part of the feed matrix. And under the conditions that are present in the intestinal tract, you've got positively charged protein and negatively charged phytic acid forming a protein phytate complex, a de novo protein phytate complex. And this has been demonstrated by a number of authors. It varies depending on the protein to phytate ratio, depending on the amino acid composition of the protein, but we can see this phytate protein complex forms very readily, particularly between the lysine, histidine, and arginine ratios in, the histidine, lysine, and arginine residues in the protein, which would typically be positively charged. That phytate protein complex is at least partially refractory, resistant to digestion with pepsin. And this is a nice segue to the things that Mike was mentioning in the previous talk, where he was exploring the relevance, the importance of adequate pepsin digestion. If you remember the antacid example that he was referring to, where you really saw a very significant depression of pepsin hydrolysis of feed, more protein moving into the large intestine, and then you get all of your volatiles and scatols and indoles and odorous compounds that come from protein putrefaction. And this has been demonstrated in a number of different in vitro and in vivo studies. This protein phytate complex resists pepsin, partially resists pepsin digestion. And the animal then, what it does is it hypersecretes pepsin and hydrochloric acid. And there's a number of examples of this, even more recent than this. This has continued to be explored over the last 5 or 10 years. Although a lot of the phytase literature today is focusing more on the beneficial effects, the pro-nutrient effects of mayo-inositol. And I don't really have much time in this talk to go into that in detail, but these anti-nutritional consequences of phytic acid, really most of the research that was on this topic was from about through the mid-90s into around 2010 or so. A lot of it was. And the animal also has a lot of extra mucin secretion. So you've reduced the solubility of your dietary protein by electrostatic interaction with phytic acid. That protein is now refractory to pepsin digestion. That stimulates an increase in pepsin and hydrochloric acid secretion, and then additional gastric and intestinal mucin in order to protect the intestinal tract from that additional acid and protease that's been secreted. So that's a few examples from the literature of just evidence for the cascade that I've just mentioned. So that's all happening essentially in the gastric gut. And then when you get into the small intestine, what we can actually measure then with phytic acid is an increase in the flow of endogenous amino acids, with obvious synthesis energy costs, maintenance energy requirements. You've got increased likelihood of putrefacted bacteria, microbiota in the hindgut, using that as a substrate, which is undesirable. So we also see compromised amino acid uptake, which is at least partially associated with sodium repartitioning, which I'll come to in a second. So we get reduced absorption both of dietary, but in particular this increase in endogenous amino acid loss. I'm very happy to share these slides with anyone who would like to see them, at least a PDF version, if you want to look at the different citations. I realize I'm going through it probably too quickly for you to read everything. Sodium, so you can imagine in the small intestine now, you've got coming from the gizzard or stomach, you've got this slightly undigested, poorly prepared protein phytate complex with a lot of extra hydrochloric acid and proteases entering the duodenum, and then the animal has to buffer all of that extra acid by producing sodium, secreting sodium bicarbonate into the small intestine to raise pH to a level that is appropriate for the pancreatic enzymes to function properly. So there's a phytate draws sodium into the small intestinal tract, repartitioning that away from sodium-dependent transport. And when you put phytase into the feed, and you reduce all of those, that whole list of things is reduced, you can see the requirement for sodium being reduced, less sodium bicarbonate being secreted into the intestinal tract, and therefore the animal's requirement for sodium goes down and it has more sodium available for other things. And that's one of the reasons why when we start superdosing phytase, you can sometimes see wet litter problems, partially through a calcium axis and partly through a sodium axis. And that's why this whole area of phytase matrices, as nutritionists, there's quite a lot of importance on which numbers do we use. Make sure that you've got your calcium, phosphorus, trace mineral, sodium matrices in place, not necessarily only economically motivated, but in order that you don't see electrolyte imbalances in the intestine and water resorption problems. So that's the antinutritional cascade of phytic acid. It starts with the electronegativity of phytate, its capacity to interact with protein and reduce solubility through the intestine, and all of these animal responses to that. Now, I'm not going to go into detail here on lots of the sort of chemistry, but I just wanted to give you a short list of things that alter the characteristics of phytic acid and may either increase its antinutritional effects or decrease the antinutritional effects. And I've listed a few of them here, things like dietary cation concentration, the ratio between phytate and protein, and I've just put this small graph in the bottom right-hand corner. This is a in vitro study looking at, I think it was protein from canola or rapeseed, one of the grain legumes. I forget now which one it was. And what you're seeing here is the phytic acid to protein ratio on the x-axis, and then turbidity of the protein phytate mixture on the y-axis, and turbidity. And what you can see here, as you increase the phytic acid to protein ratio, you get a reduction in solubility. So there's an increase in turbidity as you increase that phytic acid content. But then you reach a certain point here, and if you continue to increase phytic acid concentration with a static protein content, you can actually see the solubility being restored. And the reason for that is that you can imagine the protein becomes saturated with phytic acid such that it's essentially all of the positively charged residues in the protein have phytic acid associated with them. And then you get an almost self-repelling property. And we published that in the Journal of Agriculture and Food Chemistry. We showed that on a number of proteins that you see this very consistent relationship. So then you begin to think, okay, in my diet, what is the phytate to protein ratio? How much phytase do I need to add in order to get below that threshold level and make sure that my protein and my phytate will be soluble? And then there are other effects, protein type and temperature and so on. I don't have time to go through all of these mechanisms. In most cases, these are probably relatively minor contributors. I think the main two that you need to consider important would be the cation concentration of the diet, including calcium, zinc, and copper, and how much phytate is in that feed. So one quick comment on the phytate protein binding that I thought was interesting is that it's very much dependent on the isoelectric point of protein. Now I'm going to try and make this as simple as I can, and I've just got a few here in this box, which I'm just going to introduce them all and then just mention what I wanted to say. So as phytic acid is floating through the intestinal tract, getting more and more negatively charged as it goes, the protein that is moving through the intestinal tract is not static in its charge. And at low pH, most of that protein will be positively charged. But as your protein moves into the small intestine, it loses the positive charge depending on the isoelectric point of the protein. And you can see proteins in navy bean with a pK of 4.5. So at a pH above 4.5, navy bean protein would become also either neutrally charged or negatively charged, and therefore it won't interact with phytic acid in the small intestine, only in the gastric gut. In soybean, glycinin in soybean, again, fairly low pH. But as you come through to some of your cereal proteins, so things like caffeine from sorghum, zein from maize, and gliandin from wheat, you can see much higher pHs here. So that means that those proteins would still be positively charged even in the large intestine. So the antinutritional effect of phytic acid, the capacity for phytic acid to interact with protein, will be persistent for cereal-based proteins, but may be transient or restricted to the proximal or gastric gut in the case of protein from some of these grain legumes. So potentially in a finisher feed, where you've got much higher concentration of cereals and lower concentrations of soybean, the antinutritional or the capacity for phytic acid to reduce protein solubility and have all of these negative effects on gut health is actually increased, potentially increased, all else being equal. Oh, one more on rye. I'm going to move toward the end of my presentation over the next few slides. I just wanted to walk you through a couple of literature reviews that we published back in 2017, 2018 on the amino acid effects of phytase, which I think, again, thinking about what Mike presented before about the importance of minimizing protein flow into the hindgut, this whole question about enzyme matrices, amino acid matrices for enzymes becomes relevant. The first thing that we notice when you look at the amino acid effects from phytase is that those matrix values that companies assign to phytase typically mimic the amino acid composition of mucin and pepsin. So that ties very much back to what I was describing earlier, where the antinutritional effects of phytic acid are resulting in an increase in pepsin secretion and mucin secretion. So the contrast when you put phytase into the feed and you're reducing pepsin and mucin flow, you get amino acid benefits that are proportionate to the amino acid content of those endogenous proteins. The literature shows amino acid responses to phytase of somewhere between 0 and 6%, typically disproportionately favoring threonine, glycine, serine, and methionine and arginine matrices are typically not so high. We also see energy effects of around 30 to 60 kilocalories. A lot of these benefits come from a reduction in the anti-nutritional effects of phytic acid, and not from a direct contribution of phytic acid to energy metabolism per se, although there may be some benefits of myo-inositol on energy and one or two other areas. So if we just take a quick look at these, I'm not going to go through all of these papers in any detail, but the two meta-analyses that we published a couple of years ago, one on poultry, one on swine. A couple of key take-home messages I just want to highlight, which I think are relevant and potentially useful. So these were the contributing papers that we reviewed. There were some papers that were duplicated in conferences, which we tried to capture, so we didn't double account for anything. But fairly large body of work that had been published showing the effects of various different phytases on ileal amino acid digestibility in poultry. This was the overview of the results that we found. So what we have here on this map is, or in this graph, is on the y-axis, the change in amino acid digestibility relative to the control. On the x-axis, the amino acid, and the blue bars simply show you the mean minus one standard deviation. So we thought if we just simply publish the mean effect of phytase on amino acid digestibility, by definition, then 50% of the time, that's going to be an overestimate of what you would actually see in practical diets. So we subtracted one standard error, and then we were able to say in about 85% of cases, you would at least get these kind of numbers. And you can see straight away the effect on methionine very low, the effect on cysteine, on threonine rather high. Very much ties back to the amino acid composition of pepsin and mucin. So this is not really coming from dietary protein digestibility, or not imbalance at least. Another interesting observation from that same work is that when we looked at the amount of phytate that was destroyed by the dose of phytase that was used in these different papers, and here we've just categorized it into essentially very low destruction of phytate, moderate through to high, where we're getting up to, in some of these papers, up to around 40–45% phytate destruction. The percent change in amino acid digestibility is very much correlated. So essentially, the more phytate you can get rid of, probably the more quickly, the more amino acid benefits that you then get. Bear in mind that a lot of these papers were published in the 1990s with first-generation fungal phytases. So I think, if anything, this would be an underestimate of reality today. And we also did some simple analysis where we looked at the anti-nutritional effects of phytate. And what we've got on this graph is how much phytate in all of those studies that I've just shown, how much phytate was in the control diets, because that just varied naturally with the different ingredient mixes that the different scientists had used in their studies, from around, well, there was some very low phytate diets and some sort of semi-synthetic diets, all the way up to fairly high phytate contents. And then just looked at the amino acid digestibility in the control groups, and we saw a statistically significant linear reduction in amino acid digestibility in the control. This is phytases to one side, just simply what is the effect of phytate on amino acid digestibility. So we were able to confirm, at least statistically associate phytate content with amino acid digestibility. There are confounds in here. High phytate ingredients are often high fiber, poorly digestible protein sources. So I don't want to overstate this, but it was at least statistically confirmed. And then when we did the same thing here, essentially the same data size in pigs. So that was the poultry data. Different set of papers, similar number. I'm not going to go through the detail of the swine results. I do show you this, which is the same picture that we saw in poultry. But what I wanted to do was show you one interesting graph, which compares the relative effect of phytase on amino acid digestibility in pigs versus broilers. So on this graph, on the y-axis, we have the percent change in amino acid digestibility with phytase and broilers, and then on the x-axis in pigs. So the first thing you can notice is that there's a difference in the quantity of amino acids that are being liberated. So in pigs, around 3% increase in digestibility, whereas in broilers, it's around 4%. So broilers perhaps more sensitive to the anti-nutritional effects of phytic acid than is the case for swine, which I think is likely to be the case. But in addition to this sort of quantitative effect, you've got a qualitative change in amino acid matrices. And I want to point out cysteine as one example, where the effect of phytase on cysteine digestibility in poultry, or in broilers specifically, is around 6%, just over 6%. But if you look at the amino acid effect, the cysteine effect in swine, it's only around 1.2%. So there's a huge species difference in the capacity of phytase to improve cysteine digestibility based on those 27 papers in pigs and around 28 papers in broilers, where we really need to be quite careful when we're applying amino acid matrices for phytase. You can't simply take your poultry matrix and transcribe it over into a swine matrix, because your sulfur amino acid assumptions would be really wrong. The sulfur amino acid effects of phytase in broilers coming disproportionately from cysteine in both species, very small responses in methionine. So you really need to have a species focus or a species dependency in your matrix assumptions. I'm going to end my presentation with just a couple of slides on a study that I published with Ravi Ravindran back in 2009, I think it was, or so, 2007, where we actually fed phytic acid to animals and looked at this endogenous loss consequence. And this is just one of the pieces of evidence that's been generated over the last 10 or 15 years, really exploring the anti-nutritional potential of phytic acid. So this is the 2007 paper, Cowieson and Ravindran, British Journal of Nutrition paper. We fed a basal diet, which was phytate-free, sort of semi-synthetic diet. It was based on casein and cornstarch and so on. That's the blue spheres. And then added phytic acid. I think it was sodium phytate we used, if I remember correctly. So again, this is a semi-synthetic in vivo trial. The dark purple is the phytate, the diet that contained 4 grams per kilo phytate phosphorus. And then we measured specifically endogenous amino acid flow in the ileum. And that was done by size separation. So all of the protein, all of the peptides that were in the diet were enzymatically hydrolyzed to below a certain molecular weight. And then when you take the digester, and you can then extract all of the peptides from the digester, and all of those above that molecular weight threshold can be categorized as being of endogenous origin, not dietary origin. So it's a really nice model that they developed at Massey University in New Zealand. So what I want to just, a couple of quick observations. Here we've got all of the different endogenous amino acids on the x-axis. This is the flow of those endogenous amino acids expressed in milligrams of amino acid per kilo of dry matter intake. And you can see when phytic acid was added to the feed, total endogenous amino acid flow was increased by almost 90%, endogenous nitrogen flow by around 80%. And then you can see here serine, 150% increase in endogenous serine flow, 135% increase in endogenous threonine. So what you're actually seeing is 2 things. One is the animal is having to secrete a lot more endogenous protein into the intestine, which will have a very significant net energy consequence. But also the whole composition of that endogenous protein is changing. You can see here we've gone from endogenous protein, which was 8.3% aspartic acid, 7% threonine. And then when phytic acid is ingested, you've now got endogenous protein that's 9% threonine. So the whole amino acid requirement of that animal is fundamentally altered when phytic acid is ingested because of the specific effect that phytic acid has on pepsin, on mucin secretion. So you've got really quite a significant change in all of these endogenous proteins with obvious synthesis costs and also microbiome fermentation potential. And then you put phytase in. This was a study with, I think it was Phyzyme XP from DuPont or Danisco at the time, 500 units of phytase. And we saw a reduction. It wasn't restored to the level of the control diet. This is a high phytate example, and 500 units of phytase is relatively little phytase, at least by today's standard. So I think today you could get closer to a dephytinized control with a healthy endogenous protein flow, but we were still able to significantly move the needle. 20% reduction in endogenous amino acid flows, 86% reduction in endogenous cysteine flow, 75% reduction in endogenous threonine flow. So now you can see why the amino acid matrix for phytase is the pattern that it has. It's because it all links back to the electrostatic capacity of phytic acid as an antinutrient and its influence on endogenous loss, endogenous protein flow. So phytic acid is, there's no question, a potent antinutrient. It all comes down to this electronegativity that phytic acid has at the pH in the intestine, and that electronegativity increases as the feed moves from the gastric gut into the small intestine. The concentration of phytic acid varies in diets and ingredients, partly by genetics, partly by fertilizer regimes, and partly due to different levels of reactivity, where the phytic acid is present, whether it's involved in a fiber matrix, whether it's locked up in different salts and so on. When it's ingested, that phytic acid interacts with cations. I've not talked much about calcium and phosphorus because I was aware Roselina would be talking more about that. But for sure, the basic amino acids, the lysine, your histidine, your arginine, interacting with phytic acid, reducing solubility and bioavailability. And that phytic acid protein complex has the capacity to generate an increase in endogenous pepsin and mucin flow, which has a detrimental effect on nutrient partitioning, sodium repartitioning, and so on. So it's a simple molecule, superficially, mayo-inositol, 6 phosphate groups, but this negative charge that phytic acid has gives it the most complex opportunities to interact with positively charged nutrients in that matrix. And it causes all these problems for us. And we've been, as nutritionists, skirting around that issue in lots of different ways, putting lots of calcium into our feed, putting more amino acids in, altering our sodium requirement assumptions. And now that we've got to a point with phytase technology where you can, in a cost-effective way, you can almost completely dephytinize the feed, we need to rethink how we're approaching some of these questions. So I hope that was useful. That's the end of my presentation. If there's time, I have no idea how much time I've taken, so please reprimand me if I've strayed. I can take a question, or we can leave it till the Q&A. I'll stop sharing.
I think we have time for one question now, and we'll have, because we have many questions, we're going to let the other questions for the section. But let me see. This is a question for you, Aaron. La voy a decir en español para la audiencia. La superdosis de FITASA actúa sobre fitato reactivo y también sobre el no reactivo. You will take the question now?
I did, yes. The translator is doing an exceptional job, so thank you, Lords. Yes, this phytase superdosing will act on both the reactive and the non-reactive phytate. But what I would say is that the sort of non-reactive or non-participatory phytate is often non-reactive because it would be insoluble in the small intestine. So what you're actually doing with a phytase superdose is you're moving the site of phytate hydrolysis more proximally. So you're getting more of your phytate degraded in the gastric gut, where more of the phytate will be reactive and soluble. So what you're doing is you're preventing that less soluble phytate from reaching the small intestine, where it becomes very much more difficult to degrade.
Thank you, Aaron. Bueno, vamos ahora al descanso. Regresamos a las 11:45. Gracias. Bueno, es momento de regresar, ya son las 11:45. Pues me toca ahora introducir, es un honor, a la doctora Roselina Ángel. Ella tiene un currículum bastante extenso, si me pusiera a platicarlo aquí se nos va toda la mañana, pero básicamente ella tiene un PhD de la Universidad Estatal de Iowa, después fue gerente de investigación de Purina Mills, y después entró a la academia, a la Universidad de Maryland, como profesora asistente, y ahora es profesora completa. Ha asesorado a muchos estudiantes de maestría y doctorado, ha tenido muchos premios por parte de la industria avícola y también por parte de Poultry Science Association. Y sin más, también quiero comentar que agradecemos a Novus el patrocinio de la doctora. Y por favor, doctor Ángel, si puede ya comenzar su plática. Gracias.
¿Ya me oyen?
Sí, sí.
Listo. Bueno, lo primero es darle las gracias a Amena por la invitación. Y estaré hablando en español si es que soy capaz, porque he estado oyendo las conferencias en inglés y ya se le confunde a uno el inglés con el español. Lo otro que les digo es que estoy con máscara porque estoy en una oficina con otra gente. Entonces tengo que ponerme máscara. Bueno, a ver si me sale la presentación aquí. Voy a hablar en general de calcio y fósforo y fitasas y qué interacciones hay entre ingredientes que nos contribuyen calcio a la dieta con la eficacia de la fitasa, la digestibilidad del fósforo y dónde estamos en predecir el impacto de la fuente de calcio sobre la digestibilidad del calcio de esa fuente y la digestibilidad de fósforo en presencia o ausencia de fitasa. Entonces quiero cubrir dos áreas realmente y le doy las gracias al doctor Cowieson porque hizo una introducción súper buena sobre los factores químicos del ácido fítico. Realmente son esas interacciones que él cubrió a fondo las que explican muchas de las cosas que yo les voy a mostrar ya desde un punto de vista más aplicado. Entonces, lo primero que yo tiendo a hacer, y yo sé que algunos de ustedes ya han visto parte de esto, pero para mí es importantísimo que entiendan el impacto que tiene el calcio sobre la digestibilidad de fósforo y la eficacia de la fitasa. Es algo que al ser las fuentes de calcio, como el carbonato de calcio, tan baratos, la gente no le pone atención. Y les voy a mostrar porque yo sigo trabajando con la industria en casos de niveles altos de calcio en dietas que ven los animales. Y vamos a hablar un poquito de las características de fuentes de calcio y por qué todas las fuentes de calcio no son iguales en su impacto sobre la eficacia de la fitasa y la digestibilidad del fósforo. Entonces, esto es un trabajo ya del 2018 que les quiero mostrar, y ya lo había mostrado anteriormente, pero hay una relación bastante buena con un R cuadrado del 0.81 en una dieta maíz soybean meal, donde le agregamos un carbonato de calcio para subirle el nivel de calcio a la dieta y se lo subimos hasta 1.11. Queríamos ver a ver si esta relación era saturable y lo es, pero hay una relación negativa entre el nivel de calcio en la dieta proveniente del carbonato de calcio y la digestibilidad aparente del fósforo a nivel ileal. Si miramos esto, es saturable porque pasamos de cierto punto en la cantidad de calcio que viene del carbonato de calcio y no tenemos más efecto en esta interacción entre el calcio y el ácido fítico. Y esa saturabilidad está basada en la relación de calcio a ácido fítico. Es decir, en dietas altas en ácido fítico, la saturación ocurrirá a niveles más altos de calcio. Y en dietas bajas en ácido fítico, esa saturación ocurrirá en dietas con menos calcio. Pero este efecto es muy, muy importante. Y lo que les quiero mostrar rápidamente es, los pollos les están diciendo a ustedes en el campo, cuando tenemos problemas de campo muchas veces, ¿cuánto calcio me estás dando? Y lo que vemos muy típicamente en dietas comerciales es que la formulación que se está haciendo está con niveles de calcio más arriba de los requerimientos. Ese es un problema inicial, pero el problema más grande es que no solamente estamos formulando por encima de los requerimientos, sino que los animales están viendo unos niveles de calcio en la dieta más altos de lo que nosotros estamos formulando y pueden ser entre 10 y 40% más altos. La realidad es que todos debemos estar tratando de que lo que el animal se coma sea lo que se formuló. Y en el caso de calcio es donde vemos esas diferencias más grandes. Y esto es simplemente para que vean un caso de comercial, en una planta comercial. Estas son dietas de retiro, donde se estaba formulando el calcio total a 0.78. Eso incluye una matriz de FITASA de 0.15. Es decir, que el calcio analizable de esa dieta es el calcio total menos lo que contribuye la matriz de FITASA, porque esa contribución no es un calcio analizable. Y como verán, solamente hay 1 de 300 muestras que se hicieron en un año en estas dietas de retiro, en esta fábrica de piensos, solo una que estaba justo al nivel formulado. El resto estaban muy por encima, a niveles hasta de 1.25 de calcio, cuando debían haber estado a 0.63. Entonces, si miramos ese efecto negativo que tiene el exceso de calcio, como lo vimos ahora a medida que yo le aumento el nivel de calcio proveniente de carbonato de calcio a una dieta, me baja la digestibilidad de fósforo. La mayoría de los trabajos que se han publicado determinan el efecto del calcio sobre la digestibilidad del fósforo basado en añadirle calcio de carbonato de calcio. Eso es lo que se ha hecho y asumimos que todo el calcio es igual. Y nos devolvemos a lo que habló Aaron tan claramente, que son esas interacciones dependiendo de cómo de reactivo sea, por ejemplo, el macromineral como el calcio. Si es soluble, es reactivo. Y si estamos en una parte del intestino donde ese ácido fítico es muy reactivo, tenemos unas quelaciones muy, muy rápidas. Entonces hay unas implicaciones muy grandes sobre entender qué es el efecto del calcio basado en la fuente. Porque la fuente del calcio impacta cómo se va a afectar la digestibilidad del fósforo. Las características físico-químicas que ya están muy bien descritas por Aaron, de la fuente de calcio usada, definen en gran parte la quelación del calcio de esa fuente con el ácido fítico. Y es la rapidez de esa quelación. Entonces, cuando yo tengo una fuente de calcio que se solubiliza muy rápido, se vuelve reactiva y tiende a interactuar con el ácido fítico antes de que la fitasa logre quebrar algunos de los enlaces de fósforo con el inositol ring. es ese calcio reactivo que tiene la mayor importancia y nos va a afectar la digestibilidad del fósforo fítico, la eficacia de la fitasa y también la digestibilidad del calcio en la fuente de calcio. Entonces, realmente esto fue una sorpresa para mí hace 4 o 5 años cuando hicimos este trabajo que les voy a mostrar, porque para mí realmente el calcio era calcio y no lo es, desafortunadamente. En este experimento espero poderles mostrar el impacto de la fuente. Se usaron 2 fuentes de calcio, carbonato de calcio, que en este caso tenía 38.6% de calcio y tenía un tamaño de partícula promedio, un diámetro medio geométrico de 0.15 milímetros. Cogimos harina de hueso de harina de carne de cerdo, le sacamos el hueso antes de que la molieran y molimos ese hueso para que tuviera un tamaño medio geométrico de 0.15 para que no hubiera impacto del tamaño de partícula. En este caso teníamos 21.5% de calcio por análisis y 10.6% de fósforo. Las 2 fuentes fueron incluidas para lograr un 0.7 de calcio en la dieta final y la dieta basal contenía solamente 0.03% de calcio, era una dieta basada en maíz, tenía 0.23% de fósforo fítico, que es un promedio que se ve en las dietas maíz, harina de soja en general, y tenía un 0.29% de fósforo total. Y se incluyó fitasa a 0 o a una dosis alta. Lo que yo quiero mostrarles, si logro quitar esto de aquí, porque es que este experimento tiene un factor que confunde un poco, porque al meter la harina de hueso también metemos fósforo. En el caso del carbonato de calcio no se mete fósforo a la dieta, refleja el nivel que había en la dieta cuando le metimos el carbonato. Pero el nivel final de calcio en las 2 dietas es muy parecido por análisis, 0.71 y 0.72. Entonces miremos el efecto de la fuente de calcio sobre la digestibilidad estandarizada del calcio de la fuente de calcio. Y si ustedes miran, cuando yo uso harina de hueso, una dieta sin fitasa y una dosis alta de fitasa en la misma dieta, no vemos un efecto en absoluto, que quiere decir conceptualmente que cuando tenemos un nivel alto de una harina de carne y hueso, la liberación de calcio por la fitasa no va a ser muy alta, porque no hay efecto, con o sin, con aquí y sin fitasa. Pero cuando tengo una dieta con carbonato de calcio en azul, vemos que tengo casi 9 puntos de digestibilidad, que se aumenta ese calcio cuando yo le añado fitasa a la dieta. En el caso de fósforo, acuérdense que tengo un factor que confunde un poco porque tenemos fósforo que viene de la harina de hueso. Pero si me meto a la harina de hueso inicialmente, tengo una digestibilidad del fósforo sin fitasa en esta dieta de harina de hueso con maíz de un 50.5%. Le añado fitasa y aumento la digestibilidad en gran parte porque hay un aumento en la digestibilidad del fósforo fítico, de la parte que viene del maíz. Pero si ustedes ven, por ejemplo, cuando yo tengo esa dieta basada en maíz, tengo una digestibilidad muy bajita del fósforo, del 14.1%, y le añado fitasa a una dosis alta y llego a un 60.8%. Entonces, el incremento del impacto de la fitasa sobre la digestibilidad de fósforo es mucho más alta cuando mi calcio viene de un carbonato. Y esto para mí fue abrir los ojos y se devuelve un poco a lo que habló Mike Bedford y Aaron, pero cuando nosotros hacemos solubilidad de las fuentes de calcio, vemos, por ejemplo, que nosotros estamos haciendo una solubilidad a 3 puntos en el tiempo para pollos, a 5, 15 y 30 minutos. Se usa una solución tamponada para poder mantener el pH de la solución a 3. Y vemos que hay un efecto de tiempo sobre la solubilidad del carbonato de calcio. Pero en el caso de la harina de hueso, no vimos un efecto significativo de tiempo. Y esto nos dejó preguntándonos realmente qué es lo que estaba pasando, porque realmente en la harina de hueso tenemos una digestibilidad mayor tanto del calcio como del fósforo. Y la realidad es que le metimos pepsina a esta solución tamponada a un pH de 3 y vemos el aumento en solubilidad asociado con el tiempo, que no lo habíamos visto en ausencia de pepsina. Y es porque el calcio en la harina de hueso está atrapado en una matriz orgánica que tenemos que destruir con pepsina, que es lo que ocurriría a nivel del proventrículo y molleja. Entonces miremos un poco los carbonatos y unos datos nuevos que tenemos como para entender qué son estos efectos del carbonato. Cuando hablamos tanto en pollos como en gallina, la fuente mayor de calcio en la mayoría de nuestras dietas, especialmente si son vegetarianas, es carbonato de calcio. Entonces se vuelve un componente súper importante cuando estamos tratando de entender esas interacciones del calcio reactivo, de la fuente de calcio con el ácido fítico y la digestibilidad del fósforo en presencia de fitasas. Sabemos que hay varios factores que afectan la digestibilidad del calcio en el carbonato y el impacto sobre la digestibilidad del fósforo en la dieta a la que se le añade ese carbonato. Tenemos claridad en este momento y les mostraré unos datos que la geología, que implica dureza, tenemos un mármol o tenemos una tiza realmente. Esta que la ven aquí es llena de poros, perdón, está llena de poros, es muy suavecita y realmente se solubiliza muy, muy rápido, no importa el tamaño de partícula. Si tenemos un mármol es muy duro, tiene una solubilidad más lenta y hay una relación entre geología, tipo de roca que tenemos y solubilidad a los mismos tamaños de partícula. La distribución y el tamaño de partícula del producto de carbonato de calcio que usemos en pollos va a ser muy importante, pero no les voy a mostrar porque no es el único factor. Y la solubilidad dinámica es muy importante también, no solamente a un punto, sino en 3 puntos distintos. Perdón. Entonces, si miramos, por ejemplo, en general, cuando el carbonato tiene una geología similar, es decir, en este caso, tenemos 2 carbonatos, son exactamente el mismo. En mi laboratorio lo bajamos a una distribución normal, pero con 800 micrometers de tamaño medio geométrico. Y exactamente el mismo bache se usó y se generó un tamaño medio geométrico con una distribución normal de 150. Y ven la diferencia en solubilidad. Esta solubilidad con la misma geología está directamente influenciada por el tamaño de partícula. Pero en general, comercialmente, ese no es el caso. Entonces, puedo tener unas diferencias en la geología de los carbonatos que pueden resultar en solubilidades similares a tamaños medios geométricos muy distintos. Entonces, a mí me preguntan con mucha frecuencia, ¿cuál es el tamaño de partícula mejor? Va a depender de la geología y de las características geológicas y de solubilidad de ese carbonato específicamente. Entonces, no hay una respuesta perfecta. El punto clave aquí es que las diferencias en geología de la roca calcárea pueden resultar en solubilidades dinámicas distintas, aunque el tamaño de partículas sea similar. Esto quiere decir que no podemos caracterizar un carbonato solamente basado en su tamaño de partícula o en una solubilidad a un punto específico. ¿Qué sabemos hoy sobre esta relación entre la geología y la solubilidad? Hemos estado trabajando con una compañía que produce carbonatos, que tiene minas de carbonatos, tanto para el uso en otras industrias, como para el uso agrícola, para el suelo, como para el uso en animales. Y ellos miran mucho las características geológicas en sus minas. Entonces, cogimos unos carbonatos en que se definieron como mármoles y miramos la solubilidad a 3 puntos distintos. Y van a ver que la solubilidad a 5 minutos es muy parecida, un poco distinta a 30 minutos, pero todos encima de 80%. Cuando me meto a lo que se llama comercialmente limestones o carbonatos que tendemos a usar, hay desde suaves hasta muy duros o cristalinos, y esto está basado en el tiempo y la presión que se ha ejercido sobre esos depósitos geológicos. Y lo que va pasando es que pasamos de unas calizas o unas tizas muy suavecitas a más duras hasta que llegamos a mármoles. Y vemos que hay una diferencia grande en la solubilidad, especialmente a 5 minutos. Entonces, en este caso, tenemos lo que llamamos tizas o calizas con una solubilidad muy alta a los 5 minutos, una variación entre más o menos 47% en estas muestras hasta 76% en los limestones, dependiendo si son suaves o ya más cristalinos, y los mármoles, que son todos muy parecidos a 5 minutos. Es una roca más difícil de solubilizar. Y esto es una área nueva que estamos investigando para determinar por qué tenemos digestibilidades distintas en carbonatos de calcio. Entonces, si miramos, en este momento tenemos más, pero les estoy mostrando 26 carbonatos de calcio que hemos hecho en la Universidad de Maryland, y van a ver que pasamos desde un 19% realmente de digestibilidad de calcio hasta un 68% en digestibilidad de calcio en otros carbonatos. Entonces, la tendencia comercial es usar un promedio, y yo les diría que yo personalmente no usaría un promedio en este caso, porque el mínimo es 19.9, el máximo es 68. ¿Con qué confort voy a usar un promedio? Y ese es el problema con el carbonato de calcio, que es la fuente que más se usa de calcio en las dietas hoy en día, especialmente porque al usar niveles altos de fitasa, estamos sacando muy rápidamente todo el fósforo inorgánico, fósforos de fosfatos monocálcico, dicálcico, y eso hace que la mayoría del calcio venga de los carbonatos. Esto yo creo que para mí es lo más importante. Si se los dejo a ustedes, los va a dejar con más preguntas que respuestas, pero este es el incremento en calcio digestible que causa la adición de la fitasa a estas dietas, que contienen una fuente de calcio que viene en un 99% del carbonato de calcio. Y van a ver que tengo un promedio de 0.04 con un mínimo de 0.01 y un máximo de 0.06. Eso es el incremento de calcio digestible. Si miro el incremento causado por la adición de la fitasa a estas dietas que contienen estos distintos carbonatos, recuerden que estas dietas son basadas en maíz y torta de soya. Tenemos un promedio de 0.14, lo ven aquí, pero tenemos ciertos carbonatos que hacen que esa fitasa no funcione bien. Y el incremento cuando tengo ese carbonato y añado la misma cantidad de fitasa es solamente de 0.06, cuando tengo en otros casos 0.18 de fósforo digestible. Entonces, si yo pongo estos números juntos, miremos el promedio inicialmente. El promedio en el incremento de digestibilidad de fósforo es 14, de calcio es 0.04. Mírenlo en proporción a las matrices que ustedes están usando. El mínimo es 0.01 de calcio, 0.06 de fósforo digestible y el máximo es 0.06 de calcio y 0.018 de fósforo digestible. Entonces es importante entender estas matrices y de dónde están viniendo con el potencial de lo que nos puede entregar una fitasa. Miremos rápidamente el impacto del tamaño de partícula. Yo se los mostré inicialmente. Este es un experimento donde tenemos 4 carbonatos de calcio que en el laboratorio los convertimos a tener un tamaño, el mismo carbonato, 2 tamaños de partícula con una distribución normal, y ven la solubilidad a los 5 minutos de 35% en el caso de 0.8 milímetros. El mismo carbonato a 5 minutos, 67% a 5 minutos. Al final, a los 30 minutos, muy parecido en un 86, casi 87%. Similarmente, diferencias entre el mismo carbonato a 2 tamaños de partícula. Bajito a los 5 minutos cuando está a 0.8 y se me sube más o menos de un 0.31 a un 0.56 a los 5 minutos cuando bajo de 0.8 milímetros a 0.15. Estos son los 5 carbonatos que hicimos en este experimento y les voy a mostrar la digestibilidad de calcio en los carbonatos de calcio y el efecto del tamaño de partícula. Entonces, en rojo está el 0.15 y en verde está el 0.8. En todos los casos, excepto uno realmente, en que no vimos diferencia en el efecto del tamaño de partícula, no hizo ninguna diferencia sobre la digestibilidad del calcio. Tenemos una interacción entre el tamaño medio geométrico y el carbonato. Por eso se los muestro así. En algunos casos una diferencia muy grande y en otras no tenemos diferencia sobre la digestibilidad del calcio. Si miro la digestibilidad del fósforo, no tengo interacción entre el tamaño medio de partícula y el carbonato. Entonces tengo un efecto entre 4 carbonatos que se hicieron en un solo experimento, donde tenemos un efecto de tamaño de partícula, de carbonato, sobre la digestibilidad del fósforo. En 2 casos es más bajito y en otros 2 casos esa digestibilidad del fósforo en la misma dieta basal a la que se le añadió el carbonato, tenemos unas diferencias de fósforo digestible muy, muy distintas. Y si miramos el tamaño medio de partícula, tiene un efecto bastante grande sobre la digestibilidad del fósforo. Pasa de 38% como main effect o un efecto primario hasta 53%. Entonces el carbonato nos puede hacer cantar la canción más linda en términos de que la fitasa me funcione súper bien, o yo simplemente al estar escogiendo un carbonato que no es el adecuado, puedo tener un efecto de la fitasa que es mucho menor. Entonces, miremos el desarrollo de las ecuaciones para predecir calcio digestible, por ejemplo. Y nosotros llevamos ya 6 años con estas predicciones. Les voy a mostrar un poquito dónde estamos y que no hemos llegado al final. Inicialmente teníamos 13 carbonatos con tamaños de partícula entre 0.15 y 0.8. Y la predicción, que estaba basada realmente en tamaño medio de partícula y solubilidad en 3 puntos distintos en el tiempo, con un R cuadrado de 0.91. Cuando le añadimos 9 carbonatos más a esta predicción, el R cuadrado se nos bajó a 0.9. ¿Qué ocurre? Y no parece tan buena realmente. Pero en este caso, para la predicción se usó el calcio en el carbonato. Eso nos da una idea del tipo de geología hasta cierto punto y del tipo de componentes que no son carbonatos de calcio en una piedra caliza. Si yo me voy ahora a... Perdón, que es que no me está funcionando. Aquí voy. Si yo me hubiera ido con estas predicciones que parecen muy buenas y las hubiera entregado como unas ecuaciones para que ustedes las usaran, para predecir calcio digestible o la cantidad de fósforo digestible que yo tendría en una dieta cuando uso esos carbonatos, sin haberla validado con muestras diferentes a las usadas, no me sirve para nada. Y les voy a mostrar. Si yo tengo mis 13 carbonatos iniciales, la predicción está en rojo y lo que se observó en los animales está en verde. Solo en un caso estuvimos justos, la predicción funcionó, pero esta predicción no sirve para nada cuando yo puedo predecir un 11% de digestibilidad y tengo un 80%. Es decir, aunque yo tengo un R cuadrado basado en mis carbonatos con los que generé mi predicción, cuando lo cuestiono con unos carbonatos distintos, si hago una predicción de digestibilidad y le pido al animal que me diga cuánta digestibilidad tiene, me doy cuenta que esa predicción no sirve. Ahora con los 22 carbonatos estamos más cerca. En verde está el observado, en rojo está la predicción inicial que les mostré aquí con los 13 carbonatos, y la predicción ya segunda estamos más cerca, no del todo, pero realmente estamos bastante cerquita a lo que se ve en el animal en esa predicción. Nos falta llegar a mejorar esto. Tenemos una mejor predicción de fósforo digestible basado en qué carbonato se use, de la que tenemos en calcio digestible. Entonces, ¿qué son los puntos para terminar esta conferencia? Si yo les digo algo es que ustedes no van a poder usar dietas bajas en fósforo, no van a poder usar unos niveles de fitasa altos y sacarle todo lo que se puede a la fitasa. Las fitasas de hoy son increíbles, nos dan unas liberaciones de fósforo, de aminoácidos, nos liberan calcio que está atrapado en ese fósforo fítico. Súper bien, pero nosotros podemos bajarle esa eficacia si no escogemos el calcio que es y si el animal está comiendo unas dietas mucho más altas de las formuladas en términos de calcio, que es lo que típicamente se ve comercialmente. Hay diferencias importantes entre las fuentes de calcio en las Américas en general, en Estados Unidos, en México, en Sudamérica. Se usan en algunas dietas todavía bastante harina de carne y hueso, en Australia lo mismo. Esas harinas de carne y hueso tienen la característica del que el calcio no es soluble inmediatamente porque está metido en una matriz orgánica. Entonces la solubilidad inicial no es muy alta, que quiere decir que ese calcio no es reactivo muy rápidamente y la fitasa puede hacer su trabajo mucho mejor. Entonces tenemos el potencial de usar fuentes de calcio que son de fuentes animales en vez de carbonatos en general, que tienen un efecto bastante negativo sobre la digestibilidad del fósforo. La digestibilidad del carbonato de calcio es súper variable. Mientras ustedes más definan tamaños de partícula, la distribución de esos tamaños de partícula, que es más importante que el tamaño de partícula, y trabajen con sus suppliers de carbonatos para saber qué geología es la que tienen, mejor van a poder usar esos carbonatos, mejor van a poder definir qué tamaño de partícula necesitan y más fósforo y más digestibilidad de ese fósforo fítico van a tener como resultado de usar las fitasas mejor. Para mí en este momento, simplemente para darles una idea, podemos predecir, basado en medidas in vitro, la digestibilidad del calcio en carbonatos. Yo diría que no con buena exactitud, nos falta. En este momento le estamos haciendo todos los análisis geológicos a todos los carbonatos en los que tenemos digestibilidad para ver si podemos mejorar esta predicción. Y en términos de fósforo, es una exactitud moderada, estamos bastante cerca, pero estamos mirando la fuente de fósforo fítico, eso lo habló Aaron un poco. Hay diferencias en la fuente de fósforo fítico, en la capacidad de ese fósforo fítico de las distintas fuentes en interactuar con un calcio reactivo. Tenemos que entender el impacto del nivel de fósforo fítico, que es súper importante y no tendemos a mirarlo tan de cerca como lo deberíamos mirar. Entonces, esos son los factores que se le están incluyendo a estas ecuaciones de predicción. Mil gracias.
Muchas gracias, doctora. Tenemos tiempo para pasar una de las preguntas de la sección de la doctora y luego vamos a proceder a ir a la mesa redonda. Un segundo aquí para organizarlas. Aquí está. Felicitaciones, doctora, por la profundidad de los análisis. Y la primera pregunta, ¿ha explorado el uso de otras fuentes de calcio como carbonato de calcio precipitado, lactato o citrato de calcio?
Hemos mirado el citrato de calcio y no vemos una mejora comparativa, porque el citrato de calcio deja de ser citrato de calcio. Ese calcio se vuelve reactivo a unos pHs muy bajitos. Lo que pasa es que el citrato no es un ácido muy fuerte, no es un quelante muy fuerte. Entonces, el citrato de calcio no nos ha dado buenos resultados conceptualmente. Hemos mirado fuentes como los fosfatos y tenemos unas interacciones muy distintas del calcio que viene en los fosfatos con el calcio que viene en los carbonatos. Pero el calcium lactate no lo hemos estudiado.
Ok, muchas gracias. Sí, Siguiente pregunta es, ¿los estudios que ha hecho con respecto a la solubilidad de calcio, ha sido siempre con la misma fitasa o ha usado diferentes fuentes de fitasa?
Hemos usado diferentes fuentes de fitasa. Entonces, todo el trabajo conceptual inicial se hizo con la misma fitasa para poder entender, entender los efectos más allá de la fitasa. Es decir, si yo lo hago con distintas fitasas, voy a ver los efectos de las fitasas y no necesariamente de mis fuentes de calcio o de carbonatos. Ya una vez que hicimos los experimentos basales para tratar de entender la fuente de fitato, la cantidad de fitato en la dieta, los carbonatos, ya se hicieron experimentos con otras fitasas.
Ok, muchas gracias. La siguiente pregunta. En el estudio que hizo de la solubilidad dinámica, que nos presenta los 3 puntos en el tiempo, ¿cómo definieron esos puntos en el tiempo?
Pues se hizo un experimento, mejor dicho, empezamos con una metodología que publicó Kuhn, el doctor Kuhn de Arkansas, en un paper en el 1999 de Zhang y Kuhn. Ellos tienen un procedimiento que usa solamente 10 minutos. Nosotros habíamos pensado que la rapidez de la solubilidad de ese calcio iba a ser importante. Entonces hicimos varios experimentos in vitro mirando el impacto del tiempo sobre la solubilidad del carbonato de calcio. Y lo hicimos en dieta en soluciones ácidas tamponadas o no tamponadas. Que el procedimiento de Zhang-Kuhn es con una solución con HCl y no está tamponada. Y encontramos realmente que nosotros podíamos tamponar sin mucho problema a un pH de 3, a un pH de 2 no se mantenía a un pH de 2 por más que tamponáramos. Entonces, por eso escogimos el 3. Y lo hicimos cada 2 minutos, en los primeros 10 minutos, y luego cada 5 minutos hasta 90 minutos. Y encontramos que en el 95% de los casos habíamos llegado a los 30 minutos, a 80% o más de solubilidad. Y encontramos que a los 5 minutos podíamos hacer el análisis de solubilidad con un coeficiente de variación entre los triplicados que lo hicimos inicialmente, que era aceptable. Mientras más rápido lo haga, realmente que sería una medición mejor, teníamos un coeficiente de variación más alto entre los duplicados y triplicados, en parte inherente porque esto es un procedimiento gravimétrico. Entonces, por eso escogimos esos puntos, basado en varios experimentos con distintas soluciones y con distintos tiempos y distintos carbonatos que nos dieran una idea dónde parar, 30 minutos, y dónde podíamos empezar que todavía nos reflejara esa solubilidad rápida del carbonato.
Ok, otra pregunta aquí. En la práctica, ¿qué se debería hacer con la fuente de carbonato de calcio antes de incluir la fitasa en la dieta?
Pues la realidad es que si yo me voy para cuando yo empecé a trabajar en la época de los dinosaurios, yo empecé en industria y en la compañía donde yo estaba teníamos claridad que entraba un carbonato de calcio, se le hacía una distribución y un tamaño medio de partícula y se le hacía una solubilidad a un momento en el tiempo, un análisis de calcio y un análisis de magnesio como mínimo. Y en mis visitas con industria en todas partes del mundo encuentro que pueden haber sido hace 5 años que le hicieron el tamaño de partícula, que están, que tienen una ficha de compra con un tamaño de partícula específico y asumen que eso es lo que están comprando y no es verdad. no le están haciendo solubilidad y en general no le están haciendo análisis de calcio, que es una tristeza porque he visto casos con problemas de campo súper fuertes cuando asumimos que tenemos un 38% de calcio en ese carbonato de calcio y nos resulta un carbonato de calcio con 32% de calcio.
Muchas gracias. Bueno, voy a convocar al doctor Cowieson y al doctor Mike Bedford y oficialmente vamos a comenzar la mesa redonda. Si pueden prender sus micrófonos también, doctores. Esta pregunta es para el doctor Bedford. ¿Son las gilanasas más beneficiosas que las proteasas para la salud intestinal?
Sorry, was the question, are xylanases more beneficial for intestinal health than proteases?
Yes, yes.
Yeah. It's difficult to say. It depends on the circumstances under which they're being used. I mean, the xylanase can influence digestibility of protein, of starch, and it can influence the amount of fermentable fiber that is being released into the large intestine. So in principle, it may sound like it would be the hands down winner, but it depends on the diet, because if the diet does not contain much in the way of arabinoxylan that can be dissolved by that particular enzyme, and if the diet has a particularly intransigent protein source, then there may well be an argument that the xylanase or the NSPase is less effective than the protease. So it's very much ingredient and dietary dependent, and perhaps even age dependent as well. I tried to point out that with younger birds, the advantage or the value of a xylanase and axos and zos addition is to accelerate microbiome development. And that's clearly something we want to do. And so the value there in the acceleration is only really present for the first 3-4 weeks. And then the acceleration value of the xylanase and the axos is diminished somewhat. So it depends on the ingredients, it depends on the species, it depends on the dietary formulation, it depends on the age of the animals.
Thanks, Mike. Otra pregunta para el doctor Bedford. ¿La lactosa se considera un prebiótico en la alimentación de aves? ¿Recomendaría su uso?
Lactose. It's a very fast burn sugar, so it's not going to get into the ceca of a more mature animal particularly effectively. One thing that I would say is that you can overfeed it in young animals and have a bloom and a very excessive production of lactic acid if there's too much being fed. So it's very much a case of a titration using the right dose. It can have some value, but it's not the first prebiotic I would consider in poultry for sure. There's better data on other oligosaccharides that I would be considering over and above that of lactose.
Thank you. Como hay muchas preguntas, voy a tratar de intercalar. Esta va a ser para el doctor Aaron. ¿Cuál es la relevancia de la constante de Michaelis-Menten, el KM, sobre la eficacia de una fitasa comercial?
Yeah, I've got it. Thank you. So in vitro, I think that there is some evidence that that KM has some impact, particularly when you're in depleted substrate concentrations. And of course, as an industry, we are increasingly moving toward depleted phytate diets, not only because we're superdosing, and I think we're getting more precise with our calcium nutrition as well. So we're moving more and more toward dephytonized feeding. But we're also using lower protein diets, which contain often less phytate. So I think in future it may be relevant. I haven't seen very convincing evidence that it's one of the main differentiators on phytase efficacy in vivo. But in vitro, in QA, QC, and phytase assays and so on, I think that it may be relevant. But obviously you want a phytase that's capable of working even in low substrate environments as aggressively as it does in high phytate environments. Both are important. So I think I would just finish by saying I think in vitro potentially, whether the extent to which that's a real differentiator in vivo, I'm not sure.
Okay, thank you. También por el doctor Cowieson, ¿considera factible que se produzca a nivel industrial una fitasa que trabaje eficazmente a pH ácido y tener una mayor degradación de fitato en la molleja?
I think that's possible. What I would say is that the soluble fraction of phytate is actually shunted into the small intestine quite quickly. So although having a phytase that is very active in the gastric gut is important, I think if it was only active in the gastric gut, you may miss a window of opportunity to continue to deal with your lower esters of phytic acid in the small intestine. So I think you want to have a pH that is a phytase that is active at low pH for sure, that can get rid of your IP6 and your IP5 very quickly, but it should also have a persistent activity on lower esters of IP4, IP3, IP2, and then work in complementarity with the mucosal phosphatases in the small intestine. So I wouldn't want to have a phytase that is super specific for low pH environments or for IP6 and IP5 only. I think there's a degradation continuity that is looked for.
Okay, thank you. Doctor Bedford, en forma práctica, al usar gilanasa más Signis, ¿cuánto puede bajar el aporte de energía de la dieta?
I think one of the issues and the problems that we have with enzyme use in the field is that we have to give a number which is going to be used robustly across a number of different diets. So in truth, the answer is in some diets, you're going to get more energy out of using azalanase than others. And generally, the value of azalanase is greatest in a diet which is of low energy content because of constraints which are brought about by the NSP itself being present, whether that's through viscosity or lack of fermentability. And in those diets, when you add the enzyme, you get a big energy increment. It's very much the same that Aaron was talking about with phytase and phytate content of the diet. You get a bigger response in high phytate diets than you get in low phytate diets. The truth of the matter is we're not measuring NSP content of our diets routinely at all. We're not measuring the proportion of soluble to insoluble NSP content, and we're not even breaking it down into the soluble fraction down into high, low, and medium molecular weight. So what we have to do is use what we have in the literature in terms of holo analysis data, meta-analysis data from the energy digestibility studies we've done and the energy extraction studies that we've done, and apply a matrix to the enzyme for each ingredient. And we all know the different numbers that people put on particular commercial products, and I would recommend you, for now, use them. We are trying to improve on that and align it to some components of the dietary fiber which we believe are assigning a particular low energy value to the diet, to wit the enzyme can address. And that is an improvement that we can see coming in the future. But right now, the way we can honestly argue that we're using NSPAs is as an insurance policy. So if you do happen to get a poor quality cereal, you're more likely to get a big bump when you use azalanase or NSPAs, whatever you're using, than when you have a very, very good quality cereal. That's evident in the literature. And what putting a matrix on the enzyme does is it enables you to extract, on average, more energy out of the diet than you would have been able to without the enzyme. It's just sometimes the enzyme is delivering that promise, and other times it's delivering and more than that promise, and sometimes it's delivering slightly less than that promise, but it's because the quality of the ingredient in your diet was much higher than you'd assumed in any case. So you get a benefit by, if you like, this insurance policy process. So if you want a number, my number's 100. I'm sure Aaron's got another number for his products, and it's based on the work that's been done on these NSPases on the specific grains that we're addressing at the moment. But improvements hopefully will be coming coming to you in the next 5 to 10 years, I would hope.
Ok, doctor Bedford, como ya dio un número, entonces, ¿cuál podría ser un número para el pollo de engorda y para un preiniciador?
Sorry, for an energy value, are you talking about?
Yes, yes.
Yeah. Again, it depends on the ingredients that you're using. I mean, if you're using if you go back to some ingredient, you wouldn't be using rye in Mexico, you're using corn. But if you're using wheat or you have wheat byproducts in there, you'd be expecting to use a bigger number. But we are using 100 kcals on our product based on the average effect of the enzyme throughout the lifetime of the animal. That's what we've been doing. In truth, there are effects that you will see in a young bird, even in corn soy diets, which relate to reduced viscosity. And there's some work from, I forget its name now, but the BASF have done some work looking at viscosity, even in corn soy diets when adding isolanase, and it correlates well with fat digestibility. So in many respects, if you have a broiler starter diet with fat in it and you're high temperature pelleting it, then the corn will create a marginal reduction in fat digestibility, which will be restored by viscosity reduction. So that's why sometimes you get these odd numbers which look higher than you'd expect in a diet that doesn't contain too much in the way of the target substrate in corn soy diets. We don't differentiate at the moment between young and old at the moment. And from my viewpoint, that's because what we're seeing is an acceleration of fiber fermentation lower down in the gut, which means that you're extracting more energy out of a young bird's diet. And by young, I mean 14 to 21 days of age. They're starting to ferment fiber, whereas ordinarily they wouldn't be getting that out of the diet in the absence of antibiotics.
Can I add, can I just chip in? Is that allowed?
Yeah,
Yes,
go ahead.
No, I mean, I fully agree with all of that. I was just thinking that in a young bird diet, quite often there isn't very much added fat to remove.
Yep.
And so you also hit some practical formulation questions around whether you can apply the 100 calories or whatever.
That's a very good point.
But you don't want to not have the enzyme in. So it's about putting it in and then bringing in the energy credit or energy matrix as you can. I think that's maybe, sorry to jump in. I welcome your input on my next question.
This is a round table, man.
Yeah, it's a round table. Yeah, I mean, actually, that's a very good point that Aaron makes. I mean, there's quite a bit of data that actually he and I were involved in analyzing, where we were looking at the generic effects of an enzyme on energy recouping from diets. And we found a really radical effect on performance of animals when added fat went below about 1.5%, somewhere like that. And the reason is it's down to the extra caloric effect of fat, particularly in young animals, where fat entering into the small intestine delays gizzard emptying dramatically, and that dramatically increases digestibility of all of the components of the diet. So if we extract all of that fat out, it's the point that Aaron's making. If I say, right, put 100 kcals on your diet, generally what you do is you take fat out and put more corn in. 2% fat, 2% corn out, in for 1% fat, for exchange for fat. And that is a disaster if the diet is very low in added fat, because you will lose that extra caloric effect and you'll lose more than the 100 kcals that you've just exchanged. So it's a very good point, particularly in young animals.
Okay. Siguiente cuesta para el doctor Aaron. ¿Cuáles serían las características de una fitasa para tener éxito con la superdosis?
Okay. I suppose with superdosing, what you're doing, because the regular dose of phytase, you are essentially getting rid of most of your IP6 and IP5. And what the superdose is doing is pushing you through that IP4, IP3 threshold and getting you down into the really low esters. So if you wanted a phytase that had been particularly selected for its superdosing utility, it's obviously a given that you need it to still be capable of getting rid of the IP6 and IP5 in the first instance, but you also want it to be able to cope with the lower esters that are being generated. So the ideal superdosing phytase might be one that can successfully degrade IP6 and IP5 very quickly, but then creates lower esters which are maximally compatible with the mucosal phosphatases so that you're generating mayo-inositol, as much mayo-inositol as quickly as you can. So I think that's probably the best answer I can give. You've obviously, I'm assuming your cost effectiveness and your thermal stability and so on, they're all a given for all phytases. That wouldn't be unique to the superdosing question.
Okay. Otra pregunta para el doctor, más bien como un comentario. Varios autores han demostrado que no hay efecto de la fitasa en la digestibilidad de aminoácidos a niveles normales de fósforo. Mucho del trabajo se ha hecho con dietas con fósforo deficiente. Entonces, si la dieta está normal en fósforo, no es necesario utilizar valor de aminoácidos. What could you comment on this, Sarah?
The question was implied. So, no, I think that's a reasonably good point. I mean, I think it's true that the lion's share of the phytase effect is driven through a phosphorus lever, and I think we maybe underestimate the extent to which that is true. However, I have seen phytase studies where adequate phosphate diets were fed, where inorganic phosphate was then added and no effect was noted, and superdosing phytase was added as an alternative treatment, and feed conversion responses were observed, and various digestibility metrics were also noted. So I don't think it's true to say that all of the amino acid benefits of phytase, or let's say the amino acid effects of phytase, disappear if your diet is adequate in phosphorus. I don't think that I would accept that completely, but I think what I would accept is that in a marginal phosphorus diet, the superdosing effects of phytase may be higher, including on amino acids. So I do think that the extra phosphorus that is coming with phytase superdosing is not a neutral participant. I think it is a helpful additional source of energy for ATP synthesis, maybe for rebalancing the calcium and phosphorus ratio, maybe for rephosphorylation of mayo-inositol systemically and so on. So I don't think it's a bystander, but I wouldn't say that in an adequate phosphorus diet, superdosing phytase has no effect, including on amino acids. But it's a very interesting question.
Thank you. This question could be for anyone, and I would like to hear all your answers, because this is very important. En la actualidad siempre usamos más de una enzima, los paquetes enzimáticos, FITASas más carbohidratos. ¿Cómo manejarían ustedes el valor de energía de estos paquetes enzimáticos?
Mike, do you want to take a first go at that one? I think it was on the energy additivity of the energy matrices.
Yeah, I think you've got to recognize that enzymes are working on the undigested material, and that's why you're getting improvements in digestibility by definition. And if you are putting a package of enzymes together which all claim on the same nutrient, then you have to recognize that as you add more and more together, then the increment that you get over and above the incumbent is going to be less than you would have got if that enzyme was fed on its own. So some degree of moderation of a combinational matrix is needed. When you're talking about phosphorus matrix, of course, there's a marginal phosphorus matrix that comes from xylanases if you really look closely at the data, but nobody's applying that to it. And you'll put the lines, you'll put everything on the phytase, and you'll never change the phosphorus matrix, and you're putting a xylanase on top of that. But it's just when the nutrients overlap. And there's fairly good data out there which actually shows the sort of the degree of reduction that you see when you're combining products. And we we do, our company, I think you're in the same boat, Aaron, we do reduce matrices on energy and amino acids when you're combining 1, 2, or even 3 products together.
Okay, any comments, Aaron or Roselina, about this topic?
One quick one from me that takes this conversation a slightly different direction is that we're not 100% sure even that energy matrices for our macro ingredients are fully additive in formulation. So I think that the dilution of enzyme matrices by 15 calories when you put a xylanase and a phytase on top of one another, and you get 120 instead of 135. The capacity that we have as an industry to really measure the impact of such an effect is quite limited. Whereas if you have a corn and a soybean diet, or sorghum, corn, soybean mixture, and you're making assumptions about the metabolizable energy additivity of those 3, you could be out by several hundred calories, and it's much more likely to have an influence one way or another. And just in the same way that we've moved as an industry towards standardizing our ileal amino acid digestibility matrices to take into consideration of this subadditivity, we're moving toward doing the same thing for energy systems. And I think once we get more precision in our formulation approaches per se, a lot of these relatively more moderate effects on the enzyme side will probably take care of themselves. That would be my thinking.
Ok. ¿Algún comentario, doctora Roselina?
Pues realmente estoy de acuerdo con los dos. Espérame aquí la traducción. La realidad es que no son aditivos. Hasta cierto punto tenemos que entender por qué se libera energía cuando se usa una fitasa o por qué se libera energía cuando se usa una carbohidrasa. Y son mecanismos distintos. Lo que pasa es que en el sistema de energía metabolizable, por ejemplo, algunos de los factores del metabolismo no se consideran en términos de qué gastos se están haciendo en términos de energía cuando, por ejemplo, en el caso de las fitasas, tenemos un efecto que baja la mucina, que tiene un efecto sobre la energía, por ejemplo. Eso es distinto a lo que va a ser una silanasa, que simplemente va a ser más disponible ciertas fuentes de energía que el animal no tiene las enzimas para digerir. Pero claramente no son aditivas. Yo creo que cuando empecemos a usar un sistema conceptualmente de energías netas, donde tengamos el metabolismo incluido, vamos a poder juntar o entender mejor cómo usar estas energías que nos traen los distintos enzimas.
Thank you, guys. Because this is very, very important, because nowadays using enzymes combo, many times there is the temptation, going really high on energy, but you know, the chickens, they don't know how to read. So they have to grow these high energy values, and they will fail in conversion. Thank you. This question is for Dr. Bedford. Mostró que las fitasas también influyen sobre la digestibilidad de la proteína del alimento. ¿Podría indicarnos el mecanismo involucrado?
I think I would refer you to my right honourable colleague's presentation. Basically, in a nutshell, phytic acid itself interferes with protein digestion in a number of ways, and Aaron pointed out several of them, but it also increases endogenous losses. And by destruction of phytic acid, not only can you improve the digestibility of some of those proteins, I was saying that phytic acid, it interferes with digestibility of feed proteins by the mechanisms that Aaron has already spoken about, which is complexing with them and preventing rapid digestion via the endogenous proteases. But phytic acid also increases the losses of endogenous amino acids, which complements or increases protein losses and protein evolution into the seeker. So it's a combination of effects. It's not a simple idea. simple solution to come up to. But it's certainly involved in protein digestion and, as a result, protein delivery into the CCA.
Thank you, Mike. Esto es para el doctor Cowieson. Hablando de superdosis de FITASA, ¿cuál sería su recomendación de la superdosis de FITASA para cerdos y pollos en dietas finalizadoras?
Well, there's a simple answer, which is the one that I have energy to give. But I think what most every company has slightly different unit definitions for phytases, and their standard dose also differs. The standard dose is typically coming in around the 0.15% available phosphorus, give or take. And then the super dose we would normally define as some factor above that, maybe 3 times the standard dose. So if you're just looking for a simple, how many units do I need to add to superdose my feed, then I would say whichever phytase you're using, ask the supplier what the standard dose recommendation is and go somewhere between 3, maybe 4 times higher than that. But then there's a complex answer, which I don't know if we have time to really go into in detail, which is whether or not how much of an additional matrix might you take for those additional units. And there's obviously an economic answer to that, which is just the point at which 1 more unit of phytase costs you more to put in than the matrix that it would come with saves you money. But you can also look at taking additional value and feed conversion on top without necessarily displacing additional nutrients. And I think the answer to that one is really a one-to-one conversation, because the feed conversion effects of phytase, I'm convinced, are also dependent on your energy density and amino acid density of the feed. And if you have a methionine limiting diet, myo-inositol and additional phytase dosing may not help you very much. So there could be a situation where it's more profitable for your business to extract as much matrix from those additional units as you can and minimize feed cost. But there's also a situation where you could look at adding some additional units, not taking a matrix on those, and transferring all of that additional nutrient, myo-inositol and so on, into lean gain. So it's not a super easy answer, but that's probably the best I can do for now.
Fair enough, a good answer. Pregunta para la doctora Roselina. ¿Sí estás por ahí, Roselina, para contestar esta?
Sí, estoy aquí.
Perfecto. Aprendimos que una relación adecuada de calcio-fósforo es 2:1, pero cuando estamos utilizando FITASA, ¿cómo se altera esta relación?
Sí, hay un dogma que habla de un calcio total de 2 a un fósforo disponible o un NPP. Aaron los definió y realmente hay unas diferencias en definiciones, pero la relación de 2:1 está en un calcio total comparado con un fósforo que ya tiene incluido algo de su potencial digestible. Ese ha sido un dogma que nos ha funcionado a través del tiempo y nos permite no cometer errores grandes, pero también nos hace no poder potenciar la formulación con fitasas muy eficaces. La realidad es que al animal, me van a perdonar el idioma, pero no le importa ni 5 qué es la relación de un total de calcio a un fósforo disponible. Al animal le interesa solamente cuánto calcio es disponible y cuánto fósforo es disponible. Y esa relación no es 2:1 y no es constante a través de la vida productiva de un pollo.
Ok, muchas gracias. Doctora, a question for Mike. ¿Cuál es la diferencia entre una enzima NSP y un estimbiótico en su modo de acción?
That's a really good question. An NSP enzyme could actually be a symbiotic. What we're talking about with a symbiotic, what we're trying to identify there, is an additive which actually sends signals to the microbiome to start producing their own xylanases. And we believe these are very short chain oligosaccharides, whether they be arabinoxylan oligosaccharides, which are known as AXOS, or simply xylo-oligosaccharides, which are ZOS. And by definition, an AXOS is a ZOS with some arabinose side chains in it, but they're not very big. 2 to 6 to 8 xyloses long, and we know that from feeding them. In some situations, just a xylanase on its own can act as a symbiotic because it produces enough xylo-oligosaccharides that they are present in enough quantities in the gut to stimulate the microbiome to change their behavior and produce more xylanases. But as I showed you, in a corn-based diet, we haven't found a xylanase yet that will produce significant amounts of xylo-oligosaccharides. In corn-based diets, xylanases will release a lot of soluble xylan from the insoluble matrix, but it's all large molecular weight material, which is not this stimulating fraction, which we're calling the symbiotic fraction. And it's interesting, what we believe is that prebiotics often, so xylo-oligosaccharides can be a prebiotic if fed at very high levels because they are quantitatively fermented. But when fed at very low levels, they have a very specific function, which is to signal the microbiome to produce their own xylanases. So the difference between an NSPase and a symbiotic is a symbiotic will produce a xylan degrading microbiome, whatever the diet you're feeding. Whereas a xylanase, its ability to become a symbiotic will depend very much upon the substrate on which it is acting. And it's not very good at being a symbiotic on a corn-based diet.
Okay, thank you. Esta pregunta es para los dos. ¿Se puede utilizar una matriz de aminoácidos de pollo en alimentos de pavo?
For which enzyme, or for any enzyme?
Any enzyme. It didn't specify the question. And use the amino acids, not phosphorus, not calcium, not energy. Use amino acids. That's what it says here.
I mean, I'll go first. Aaron, you shoot me down.
Oh.
There's so little data in turkeys compared to broilers when you do the hollow analysis, which is exactly what Aaron was describing between pigs and chickens there, where you get a much better idea of what the value is in a particular enzyme in releasing amino acids. You get a much better idea of how consistent a product is when there are multiple studies that you could draw from. The problem we have with turkeys is you're probably talking, 10% or less of the number of trials in turkeys compared to broilers on almost any enzyme, and particularly in looking at amino acid digestibility, it's very difficult to say that you would identify anything that's specifically different from the broiler or whether it's exactly the same as the broiler. But the data that is out there at the moment suggests it works somewhat similarly, but I don't know how dissimilarly it is in the way that Aaron was describing the difference between the pig and the chicken for phytase, for example.
Okay. How about you, Aaron? Some comments on this?
I think I would go along with that. The turkey diet, obviously, and the young turkey diet is a very high protein diet. So there might be some compositional effects that are independent of the species, just as a function of the different feed that we provide. But I think in general, the turkeys, we have run turkey trials with different enzymes, the protease and phytase I'm aware of, and they do work on amino acids, but as Mike said, the data size is not great, and we tend to extrapolate a bit like we do for sows and even laying hens to some degree, and ducks and things like that, some of these relatively minor species. So I wouldn't absolutely nail my colors to the mast on that one, but I don't see any reason why they can't be transferred.
Okay, thank you. This question is for Aaron. Do you think it can be used inositol directly to the feed? Would be that economically feasible and okay for poultry?
Well, you can buy myo-inositol, but it's about $20 a kilo. So I don't think it would be very economical to add it to feed. Not as economical as you can supply it via phytase use, for example. But there have been several studies done adding mayo-inositol to feed, often at the levels that you might expect to generate from phytase. So, you know, that kind of 0.1 to 0.2% mayo-inositol, which is approximately how much mayo-inositol you generate when you use phytase. And results have been generally positive, but a little bit mixed. I would say 70% of the trials I've seen where mayo-inositol was delivered orally to poultry have shown overall improvements in energy digestibility, in muscle protein accretion, and reductions in feed conversion. There's one study in laying hens that I'm aware of where there was a neutral or even slightly negative effect that's not really well understood. So I think overall it seems to be positive. But I think the mayo-inositol effects may be age dependent, and we published a couple of studies back in 2013 with Damian Józefiak's group in Poland, and in that study there was a slight negative effect of mayo-inositol on feed conversion in the starter diets up to day 14, but then it reversed and it became quite positive later on. And I wonder if it's doing something on protein accretion pathways, because we have done some gene expression work where you can see when mayo-inositol is put in the feed, you get a very significant upregulation of genes downstream from IGF-1. So that the kind of the mTOR signaling pathways that shuts down gluconeogenesis and promotes protein accretion. And that is the stage, you know, in the grower phase, in the finisher phase, where the bird is really depositing maximum muscle gain. Mayo-inositol might be uniquely beneficial there, but in the young animal, maybe it competes with glucose absorption or something. I don't know for sure, but it's an interesting molecule. It's not one we fully understand. There's also recent work showing that there might be some effects on dopamine pathways in animal welfare, which is an area that's not yet been fully explored. So the pet industry, companion animal industry, are quite interested in that. So that's probably what I would say. I don't know, Mike, if you've got anything to add on that.
Inositol. The only thing I would add is that I know you covered 99% of it, I would say, and for sure. The only thing I would add is some recent work that we've been involved in is looking at the influence of inositol potentially on as a precursor for IP5, which is used by the chicken as an oxygen shunting molecule, just like we use BPMG in human on hemoglobin. And so what effectively it's doing is increasing the ability of blood to release oxygen in very anoxic tissues. And that might be part of the rational reason why going to very high levels of phytase, increasing inositol absorption, increases IP5 in the red blood cell, which reduces to some degree the incidence of woody breast, which is associated with anoxic tissues. So as Aaron said, it's involved in so many different pathways that it's really difficult to pin your colors to one mast and say this is how it's working and this is the response you're going to get. But in answer to the question, you know, would you add it? No, as Aaron said, $20 a kilo versus how much would it cost to superdose to get a kilogram of inositol, 2 kilograms out of your current diet? It doesn't make sense.
Okay, guys. Well, I think we're going to stop here. Really appreciate your talks and your interventions. I guess we all learned many things today, as usual, when we are with you. So thank you very much, guys. Para todos, muchas gracias por haber estado aquí. No sé si el señor presidente quiere dar algunas palabras. Doctor presidente.
Les agradecemos mucho su participación. Ha sido un seminario, ha sido un simposio muy interesante. Gracias. Por favor, Aaron, por favor, Mike, si tienen que hacer otras cosas, adelante. Una disculpa por robarles estos 10, 15 minutos de más. Muchísimas gracias a nombre de la MENA.
Pues igualmente yo agradecerles a los ponentes de parte de la MENA, agradecerle a nuestro moderador, el doctor Sergio Fernández, y agradecerle a todos nuestros patrocinadores de este evento. Les dejé por ahí en el chat una serie de enlaces. Está la encuesta de este evento, si nos pudieran ayudar contestándole para saber pues la dirección y qué opinaron de este nuevo formato. Igualmente invitarlos a nuestros siguientes eventos. Tenemos el Seminario de la Industria el martes 1 de junio. Este está presentado por DSM. Vamos a tener las presentaciones del Dr. Doug Corber y del Dr. José Antonio Cuarón. Entonces los esperamos el martes 1 de junio a las 17 horas. Igualmente, pues dejarles ahí las fechas de nuestros siguientes simposios virtuales. Estos son requerimientos nutricionales en aves el 7 de junio, es igual lunes a las 10 de la mañana. La de acuacultura, presente y perspectivas a futuro en nutrición alimentos, este es 5 de julio a las 10 a.m. Obesidad en perros, causas biológicas y estrategias para enfrentarla, es el 16 de agosto a las 10 a.m. Microbioma y salud intestinal el 27 de septiembre y sostenibilidad, medir para mejorar, de concepto de intervenciones prácticas en la granja. Este está por confirmar de la fecha, es en octubre. Igual pues ya les mencionamos, ¿no? Es uno al mes, entonces en noviembre y diciembre también tendremos estos eventos, por ahí les pasamos próximamente las fechas. Entonces ya tienen ahí los enlaces de estos simposios, se pueden ir registrando de una vez. Pues les dejo los carteles de nuestros simposios, el de requerimientos nutricionales en aves, con la participación del doctor Rostaño, del doctor Mateos y del doctor Shibirao. El de acuacultura, con las participaciones del doctor Ulfred Focken, del doctor Biswas Biswajit y de Bruno Araujo. Y el de obesidad en perros, con las participaciones del doctor Jorge Lara Arias, el doctor Jorge García Rendón y el doctor Francés Villarroja Gamboa y la doctora María del Carmen Camacho Rea. Entonces, los esperamos en estas siguientes fechas. O sea, váyanse registrando. Y pues recordarles nada más que tienen ahí el canal de Amena, donde vamos a tener las grabaciones de estos seminarios, de estos simposios también. Ahí estará este primer simposio próximamente, donde podrán revisar pues, la información que se presentó aquí. Y pues agradecerles finalmente a todos ustedes por asistir a este nuestro primer simposio virtual Amena y agradecerle también a nuestros patrocinadores, entre ellos Adiseo, ADM, Elanco, DSM, Nuproxa, Bevista, Novus, Provimi y Uvefarma. Los esperamos entonces en nuestro siguiente seminario de industria el 1 de junio y en nuestro siguiente simposio virtual el 7 de junio. Muchas gracias a todos.